So the YouTube site says live in 15 minutes. Is there any way to get it to happen sooner?
Put us live, so we should be. It's showing on my screen.
Okay, let me refresh. Yeah. Okay. Sorry. Never mind. I just had to refresh. We're good. Minutes. Is there any way to get it to happen?
I think we're on, Sam.
Yeah, I gotcha. Okay, hi. I would like to welcome everyone to this meeting. It is really exciting to have so many people who are tuning in from all over the world. My name is Sam Sober. I'm in the biology department at Emory University, and I'm going to be moderating the symposium today along with my co-host, Gordon Berman. In a few minutes, Gordon is going to talk in some detail about the rather unusual format that the symposium is going to take. But before we do that, I want to talk a bit about the sort of the origins of this meeting and why this group of people is together right now. So let me start my screen share again. Talk in some detail about the rather unusual format. Okay, sorry about that. Okay, so today's meeting, welcome to the inaugural Simons-Emory Symposium on Motor Control. So this, oh wait, sorry. And I apologize once again for starting the wrong presentation. Let me try that again. Pardon me. Excuse me, this will just take 1 second. Okay, here we have it. No matter how long this seems to be taking for all of you, I guarantee it feels 20 times longer to me, but we have it now. There we go. Okay, for real this time, my bad. Okay, so finally, I'm welcoming you to the inaugural Simons-Emory Symposium on Motor Control. So this symposium is the kickoff event for a new research consortium that is sponsored by the Simons Foundation. And this consortium is the Simons-Emory International Consortium on Motor Control. As you can see from the list of investigators at the bottom here, this is an international network of labs that includes both experimental and also computational groups examining motor control. And the objective of this consortium is to create both new electrode hardware and also new analytical approaches to understand motor control across a really wide range of species and behavior. And before Gordon gets to the details of what the symposium is going to look like today, I'm just going to take a couple of minutes to tell you about how this consortium is planning to achieve this goal. So the primary, in terms of experimental methods that we're creating, the main experimental tool that we're developing are flexible high-density electrode arrays for getting really high-resolution recordings of muscle activity. So our lab at Emory has developed this technology first for recording the spikes from individual motor units within the vocal muscles of songbirds. And if you want to follow the citations below, you can see that we've used the data that come from these electrode arrays to reveal how patterns of motor unit spikes control songbird behavior. So what you're looking at on the left here, this is actually a very early version of one of these arrays. This is sort of a prototype with 16 contacts. On the right, you can see a little bit of, this is an example of a single motor unit being isolated from this array. Since initially developing that, we've scaled it up quite a bit. So what you're looking at here, this is a raw data stream from a 32-channel recording. So what you're looking at is an assortment of individual motor units, in this case isolated from the respiratory muscle of a songbird as it's breathing. So what you're looking at is an ensemble of single motor units during the generation of vocal behavior. So one of the central hardware goals of the consortium is to scale these array technologies up in 2 ways. First, in terms of channel count. So we're in the process of scaling these up to 1,000 channels, or just over 1,000 channels, and also to scale them up across species. So as part of the consortium, we're creating these for the range of species you see here. What you're looking at on the right, this is a schematic of a prototype of one of the first mouse arrays that we created. I think Rui Costa, who's the last speaker in the consortium, will show some data that we recorded, single unit data that we recorded from a behaving mouse. The other major kind of tools that we're developing are computational tools. So alongside of and sort of in tandem with creating these new electrode systems, the theorists in the project will be creating and eventually distributing these different methods for analyzing the kinds of motor systems data that we and lots of other people are generating. And then, as far as branching these out across lots of different experimental organisms and behaviors, the experimentalists in the collaboration, who are shown at the bottom here, will be applying all of these different methods in sort of a common analytical framework across this really wide range of motor actions. Another major initiative of the consortium is that we are going to run training courses that will be open to the entire scientific community. These will be courses, some will be on experimental tools, some of them will be on the analytical frameworks and methods that we're developing. Please stay tuned for announcements. We will be releasing details of when these will be happening shortly. And then finally, before I turn the mic, as it were, over to Gordon, I want to thank some really important people who have made this symposium possible. First, Jerry Fischbach and Alyssa Schaefer at the Simons Foundation. Also, as you can tell, we are Emory Theory and Emory's Theory and Modeling of Living Systems Initiative is donating lots of expertise and web hosting to us. So thank you, Tiara, and others there. And then I want to give a special thanks to Amanda Jacob, who's the operations manager of the consortium, for just doing a ton of work to put this together and make sure it runs smoothly. So enjoy the symposium, and I'll turn it over to Gordon now.
Thanks, Sam. So I think that, thank you everybody for joining us here today. This should be a fairly interesting symposium and definitely a bit of an experiment and hopefully a rather provocative set of talks to talk about. So as Sam noted, the format will be pretty unusual. And that what we'll be doing is, so we've given all of our 8 speakers today a prompt, which is what tools are we missing to understand motor control, and what could we learn if we had them? And we're giving them 10 minutes to actually give their answer to this, which is a patently ridiculous assignment. And they've all done really amazing jobs at coming up with these things. So I look forward to seeing all the really ambitious talks. And then on top of that, because of the format, there'll be 20 minutes per speaker. The first 10 minutes will be their talk, and then we'll have 10 minutes for discussion. So we'll probably start out with a few questions from people up here on the panel, which will include all 8 speakers. Plus also we have a few discussants today, myself and Sam included, but also Ilya Naimanman as well. Megan Carey was scheduled to be one of our discussants, but unfortunately she wasn't able to make it today. And then after we sort of discuss a little bit what everybody should do, if you have a question, you should ask it on the top chat and on the YouTube channel. So you need to have a Google account in order to do that, but you should ask questions. And then we have a great team of grad students and postdocs that are feeding us questions here on the Slack channel. And so Sam and I will make sure to feed those questions up to the speakers, and hopefully we can have a great discussion. So with that, let's get on to our first speaker. So our first speaker will be Abby Pearson, and she's coming to us from University of Chicago Anschutz, and her group does a bunch of really cool stuff, understanding cerebellar contributions to smooth movements, doing fancy stuff from neuroanatomy to optogenetics to electrophysiology, and really careful, cool behavioral measurements as well, which is obviously near and dear to my heart for anyone that knows my research. So she's going to talk today, and so Abby, take it away.
Well, can everybody hear me? Can I get a thumbs up? All right, great. Well, so hello out there in TV land. I guess that you're here. I just wanted to first start by thanking Sam and Gordon, as well as the Simons Foundation, for the opportunity to share my thoughts on this interesting question about tools that we're missing and what we're actually aiming for in the study of motor control. Now, this is not going to be a typical data talk for me, but rather a rumination on some of the ideas that fascinate me about motor control. Now, many of us are familiar with watching robots move. They're clunky, they're rote, they're rather inflexible, although that's changing with new technology. So much so that we use the term robotic to connote stiffness and lack of expressivity. Now, the first time I saw this kind of a robot, this is called a passive dynamic walker, I thought to myself, this is how animals move. I was totally floored. Now you can easily appreciate how naturalistic this gait looks. And yet these robots are truly passive. They have no motors, no actuators. It just uses passive inertial responses to the treadmill in this case. It uses gravity and the mechanics of its parts to move. Now, these designs are called underactuated, meaning they're not controlled along some of the trajectories. Hello? Along some of the trajectories that they move along. And so this idea of underactuated is or underactuated or intermittent control is actually seen in the field of biological control of movement. But I think these ideas are often overlooked. And so today I'm just going to be exploring these ideas a little bit more. So I think by looking at this passive dynamic robot, it would be easy to jump to the conclusion that because we move like we're underactuated, we are underactuated systems. But this is definitely not the case. When we put our minds to it, humans can do pretty much anything, anything that's biomechanically possible. We can play the Tchaikovsky violin concerto if we put enough work into it. So axiomatically, we are not underactuated systems. Rather, we seem to be something like hyperactuated systems. And yet, when we're watching any virtuoso like Midori play, it highlights a paradox, which is that they make these hyperactuated feats look easy. And I'll argue that the reason that these actions look so easy is that they're in fact are easy to the performer, at least energetically, to them because they've learned such efficient ways to produce these actions. So this paradox of immense skill and apparent ease brings us to this central hypothesis that I'm interested in seeing the field of motor control develop in the coming decades. And that is that the nervous system learns to control the body in an underactuated or intermittent way. So this brings us to my answer to the question of the day. We need tools to test underactuation, and we need those to be both sort of the things of science, but also theories of learning that better incorporate efficiency into reinforcement algorithms. So I've broken down where and how to look for these ideas in the nervous system, broken it down into the periphery and central learning roles. And so within the periphery, I think we can ask the first big question of underactuation, which is, do skills become underactuated with learning? Now, at a broad level, we know the answer to this question is yes. First, we know that co-contraction of muscles is reduced during the course of skill learning. So here I'm schematizing this transition where early in learning, there's a lot of co-contraction that stiffens the joints and leads to more explicit control. While later in learning, muscles take on more turns, the joints loosen, and muscles turn on at the beginnings and ends of movements, like in the swinging phase of a human leg movement during locomotion. So many studies support these notions at the whole muscle level. Thus, these observations are broadly consistent with what would be expected for intermittent control of passive dynamics. However, by relying on whole muscle EMGs, our view of these processes is intensely crude. And so that's really been limited by technique. And we don't know what the individual actuators of movement are actually doing, i.e. the motor units. So I wouldn't be here if I weren't excited about the technology being developed by Sam Sober and Muhini Bakir at Emory that will allow single unit motor recordings from contracting muscles. And I think with this technology, we'll be able to test key tenets of ideas of intermittent or underactuated control, predicting that over the course of learning, motor unit activity should become sparse in time and space, even as movements become faster and more precise. So this brings us to the next question, which is how might learning theories interface with underactuation? Now, the most obvious advantage of underactuation is energetics. you don't want to be controlling what gravity or inertia will do for you anyway. In fact, if you are controlling those aspects of your movement, you're wasting energy, which is why we can pretend to act like robots. But where within our motor learning theories does reinforcement of economy exist? And so here I'm going to draw a typical control systems diagram that has a controller that generates commands that control the effectors, the parts of the body that are actually moving. Now, there's a popular framework for de novo motor learning that's been explored intensively in songbirds that borrows from actor-critic architectures of reinforcement learning. And the idea in vocal learning is that there's a period of vocal exploration, which is called babbling, where outcomes are being monitored. Correct outcomes will be stumbled upon occasionally and reinforced. Then this will bias the controller to produce those patterns again in the future. Now, this idea has received empirical support from a number of labs, and most mechanistically by Vikram Gadekar in Jesse Goldberg's lab, who found that dopamine neurons in songbirds burst in response to unexpectedly good vocal output. So this is really cool, but what we're missing in these theories is a learning of economy. Now, from what we've seen about the progression of over-actuated to less actuated, the prediction would become that energetically cheap movements that accomplish the same goal as energetically expensive solutions are selectively reinforced. Now, nobody knows how this works, but I think it's interesting to note that basal ganglia circuitry that controls motor opponencies, agonist, antagonist musculature, is regulated through dopaminergic tone. When basal ganglia and dopaminergic circuits go awry, contraction and rigidity emerge, which are, of course, famous symptoms of Parkinson's disease. Now, future studies might ask whether exploration or experimentation with vigor is a component of the exploratory space of motor learning. The idea is that if motor vigor fluctuates to engage or disengage co-contracted muscles, it might allow the motor system to explore cheaper solutions to the same problems, seeking the same goal. Now, the final idea of this framework and the final component utilizes internal models, which are near and dear to my heart as a cerebellologist. So internal models are necessary ingredients in the setup of underactuation or intermittent control, because it's only at some times within a movement that intervention is necessary for control. And the rest of the time, you really want the nervous system out of the way. And so the muscles that engage at the beginnings or ends of movements outside the period of passive dynamics need to be well regulated with estimates of where the effector will be in time. Now, these ideas predict that there's a site or sites within the brain that learn continuous representations of body movements that occur both as a consequence of the nervous system moving it through forces of the muscles, but also as a consequence of the forces acting upon the body from the environment. Now, we actually know at least one site in the brain that does this. In fact, it's exactly what the cerebellum is set up to do. Purkinje neurons learn to predict kinematics of the body and the physics of the environment. And so my question with that knowledge is really, how does that continuous control get turned into an intermittent control signal that then influences the overall controller that's being sent to the spinal cord to control the body? And so that actually sums it up. I think we need better understanding of the mechanisms of learned underactuation if we really want to get to the bottom of naturalistic motor control. And so with that, I'm happy to take any questions.
Great. Thanks, Abby. That was fantastic. It looks like we have a question from Sam. Sam, do you want to ask?
Yeah, thanks. Okay, so that was fantastic. Thanks, Abby. So, okay, so you were talking about intermittent control sort of mostly from the perspective of energetics, and that completely makes sense. Like you want to minimize co-contraction to save energy. But what about sort of all the other good things that intermittent control might be for? Like maybe it goes beyond just reducing overall energy costs towards actually improving performance or dexterity, or what we see a concert violinist do. So, I mean, energetics is a fantastic place to in some ways very quantifiable, but how would we develop tools to really get beyond what intermittent control could be doing beyond simply energetics?
Yeah, I think it's a good question. So, in thinking about this, I think that one of the questions that would emerge is if we have access to these motor units at high density, we can actually access whether there are redundant solutions to the same problem. So if we know that the kinematics are identical, and that there are more efficient ways of doing it, then I think it argues more in sort of the efficiency camp. If, on the other hand, it turns out that the kinematics are always subtly improving, then I think that we can go back to this idea that there's just feedback reinforced solely. And I think it's really going to be a combination. I think that there are performance aspects that improve with under actuation without a doubt. But I think that there's still ways of doing things efficiently that can be sort of selected for independent of output or feedback.
Okay, thanks.
Great. It looks like we have a question from Amy. Amy Bastian. Amy is not a unique specifier here on our Zoom call today.
Right. We have multiple Amy, or at least two Amy's. Hey, Abby, that was a great talk. Thank you so much. My question is really about how what kind of signals reach the brain that actually truly reflect energy cost. So I think that when you think about signals, it seems that that's necessary for what you're talking about. And as far as I know, signaling from the periphery doesn't specifically carry energy cost information. It's much more indirect. So could you comment on that?
Yeah, it's a very interesting question. Like, you could imagine that if there were some sort of a cost minimization function, it wouldn't necessarily have to be through the muscles. It could be through efference copy signals that need to become sparser, for example, over time. But I think too that there are feedback from proprioceptors that would signal tendon stiffness, joint stiffness, those kinds of things that could be essentially related to co-contraction of muscles.
So just to follow on very quickly, so those are proxies for energy.
Exactly. Those would be proxies, right.
Right, so a related question, we have a question from Andrew Murray from the YouTube chat. Would energetics be estimated by the internal model, measured from metabolic signals, both?
Yeah, I think this is similar to Amy's question, which is what would this even look like? I mean, so one possibility would be that there's a cost function on the sort of intermittent controller, let's say the cerebellar output. Or sort of the consequences of the internal model. If the idea there were that the brain wants to minimize that control signal, then you just throw that into your cost function. It doesn't have to be muscles per se. I think that we could probably, come up with a lot of different proxies that the brain could have access to that are essentially these ideas. So I don't want people to get fixated on like ATP costs in the muscles necessarily, but more of the efficiency of control, even at the neural level.
So, I mean, going again with this notion of optimization. So this is a question also from YouTube and I apologize for my pronunciation. So you can send me an email telling me how I mispronounce it later, but from Simisola Aludare, who asked, can learning under actuation fall under the banner of optimization, or are these completely different concepts?
Well, I think there's some level of optimization in there. I don't know along what axis the person is asking about what's being optimized. So in some ways, I think this dovetails with Sam's question. It's like, is is an underactuated movement a better movement, either for the task to be accomplished in total or not? And so I think that there is some kind of a space of optimization that I think underactuation falls into. It's a lower energy state, probably for both the brain and the body to operate within. And in that way, I think there could be optimization, cost minimization principles at play.
So also from Spencer Wilson, is this efficiency not captured by control magnitude terms in the optimal control cost function?
I think that I would imagine that there may be, but I'm not familiar enough with those models to actually say.
Okay, fair enough. Lena, did you have a question?
Just more of a comment, and what about neural energetics? I think that's also important. And I think when we talk about cost functions and optimization, like how does that happen in the nervous system? How do you just turn on and off different costs?
Well, I think there are a number of models that use essentially feedback pathways to sorry, I just got a text message. There are a number of ways to generate in feedback circuits, essentially minimization functions. So I think that there are beautiful examples of inhibitory feedback control within the cerebellum that it sort of rides within this this sort of set point through feedback mechanisms. And so there's all kinds of architectures that I think can do this. And it's really operating in a space of neural energetics or activity minimization. And so I think that there's ways to do it.
Awesome. Okay, so we can take at least one more question. And so this is from Joshua Kashabak. And he asks, should the efficiency critic related to be related to the internal model? Also, is it possible that the efficiency critic is under the hierarchy of the actor critic?
Yeah, both. So kind of what I imagine is that the output of the internal model. So, okay, there's some sort of subtleties here. So the internal model could be used as a prediction for anything. I'm sort of thinking about how that internal model might be used to compute the timing of an intermittent control signal, say at the end of a movement. So, your arm is swinging through the air and then suddenly muscles need to be engaged to stop it. I think that could use an internal model, but it is not itself an internal model. So, so that kind of a signal could be added into the controller per se, but it could also be added into that efficiency critic. That is that critic cares about how much that intermittent controller is active, and it wants it to be active as in such a small way without generating errors. And so there's that. But then I think as far as the actor, this is kind of the whole like dichotomy between actor critic and efficiency critic isn't super it's not necessary to happen. It could be that it's all this, it's all the critic. It's all one critic. But the point is that it might be evaluating multiple components or aspects of the movement. So maybe that answers Josh's question there. Joshua.
Great. So I think we probably need to move on, but thanks, Abby. That was really cool. Our next speaker is Andrew Pruszynski. He's the Canada Research Chair at Western University in London, Ontario, and his lab does all sorts of really great work, sort of bridging neural encoding ideas from the periphery up to cortex and really thinking about these things in a deep way. And I think he's going to talk about inputs and outputs in neural systems and why we don't understand them.
Okay, can you hear me?
Yep.
All right. And can you see the screen? Great. Well, thank you very much for having me here today. I think it's going to be an exciting seminar. I'm glad to be a part of it. I'm going to emphasize a particular set of questions that are maybe a little narrower than what you've seen previously, and that is to say that in order to understand motor control, I believe that we really need to focus on the peripheral inputs and outputs, and that we don't currently have a solid understanding of what's going on there. And to some degree, that's because the experimental toolkit that's been deployed in the central nervous system has been so far underdeveloped within the constraints of the periphery. So here is just a classical picture of what the sensory motor control might look like in the context of primate reaching and grasping. You of course have motor commands descending down and sensory information coming back. And there's multiple nested feedback loops that are integrating that sensory feedback, be that intermittently or continuously in complicated ways. And in addition, there are multiple parallel pathways, both for the ascending sensory information and the descending information. And there's a lot of work to be done in understanding and picking apart how these nested feedback loops and parallel pathways interact. But for the purposes of today's talk, I want to focus all the way down here and point out that when you make commands to move your arm, you actually have to create that via activation of these individual muscles. These muscles have their own biomechanical, anatomical, biophysical complexities. They are highly different if you're talking about the pectoralis versus finger muscles. And we really need to understand the details of that actuation. Today, I'm going to talk about the other end, with also issues of what happens on the sensory end, meaning you don't really get information about how the hand moves around per se. What you get is a set of inputs caused by mechanical deformations in the various tissues of the body, in the muscle, in the tendon, and in the skin. And the case I want to make here is basically that if we don't even understand what's happening all the way out here in the bitter end of all of this, it's very hard to make inferences about what's happening in these higher order processing centers. And I want to do that via a specific example, I think, just to ground ourselves, and then I'll back out at the end and say what kind of technologies are really needed. So if you've seen my talk before on this thing, you've seen this video a lot, but here's a picture on the left of a woman who came into Roland Johansson's lab and was asked to strike a match. There's no experimental manipulation here, and clearly she can do this very easily. On the right here, we have, he has injected marcaine lidocaine in the digital nerves and removed inputs from a few thousand mechanoreceptors just in her fingertips. And what I want you to notice is how she loses her dexterity. She's unable to pick up the match, she drops it. She doesn't even understand how she should orient her hand relative to the match. She has great difficulty applying the correct forces to stabilize that relative to the matchbox. With great diligence and effort, eventually she can figure it out and succeed at this task. Now, what's interesting here is that she has everything intact. I mean, she can see, she's obviously well practiced. She knows this behavior from her real life, and she knows it from the lab just from moments before. But she has all the proprioceptors, she has all the vision, but by losing those few thousand mechanoreceptors, she's clearly not normal in the context of her hand use. So what do we know about how this kind of information is transmitted from the periphery to the brain? The general model that you'll find in textbooks is the idea that these peripheral receptive fields have this, that we've eliminated via this anesthetic, have this simple receptive field property in the sense that they look like these little pixels. They're little Gaussian receptive fields of small diameter that are basically tiled across the fingertip or distributed across the fingertip. And what you end up having is this bitmap representation of the tactile stimulus on the finger. Think of it like the visual analogy is here, like that's a retina. Okay, and now when you stimulate it with a particular spatial thing, like in this case, classical seminal work here by Ken Johnson and his colleagues at Hopkins, if you stimulate it with an A, the population of neurons in the skin will veridically represent that letter A. You can see the A in the spatial representation of these first order tactile neurons. So in this concept, these kind of periphery act as really passive wires. The veridical spatial information about tactile stimuli are sent to the brain, and the brain does that further processing. So you get some uncorrupted image that comes in here, and it's the work of, for example, the primary somatosensory cortex, where a lot of effort has been spent to figure out the features of that tactile stimulus, extract things like the orientations within those different stimuli, the direction of their movement, their curvature, and so on and so forth. But that sort of beguiles the detail detailed neuroanatomy that we've known from the 50s about what's happening here in the mechanoreceptors. And that is to say that these mechanoreceptors branch and innervate many mechanoreceptive end organs. So unlike the picture here, the cartoon, the anatomy is actually much different. And you have this dendritic-like branching all the way out at the bitter end of the skin. And in fact, if you look carefully, if you map these receptive fields in very fine spatial detail, you see that yes, some of those receptive fields look like these pixels. They have a small central spot of activation and smooth fall off, but the vast majority do not. They are spatially complex. They have multiple spots of high sensitivity. They're heterogeneous across receptor types, across neurons. And so these various little hotspots within these receptive fields likely reflect the endpoints of these mechanoreceptors somewhere under the skin, as smeared through the skin mechanics, of course. So we've been working under the general idea that this branching constitutes a peripheral computational mechanism for spatial feature extraction. And the evidence for that, I can't go through all of it, but I'll just show you one quick example. If you record from one of these neurons in the peripheral nervous system and stimulate it with various edges passing across the receptive field, what you'll see is that that neuron highly structures the response as that stimulus passes over the receptive field. So as this edge passes over the receptive field, it hits different hotspots, and you get this really precise temporal sequencing of activity. Now, critically, this temporal sequencing of activity changes as a function of edge orientation, meaning upstream observer could tell what orientation is being stimulated by looking at the sequence of neural activity. And we've gone on to show that this is highly robust across stimulation speed, direction, and it's very, very, and more recently that this really does fine resolution of edge orientation down to a few degrees differences between edge orientations. So what does this say? It really says that there is some computational power already here out in the skin. These dendritic-like branches of these first-order tactile neurons are already removing or filtering or optimizing the kinds of information that these upper centers, will further refine and further compute on, but they're not computing on a raw image per se. They already have been pre-filtered in a very specific kind of way. And that means if you want to understand what these upper levels are doing, you need to take into account that the periphery has already sent some very specific structure in that input space. So what might this be good for? I don't have time to get into it, but we think that this particular kind of structuring is absolutely critical for these kind of behaviors that require very precise hand control, because this temporal structuring, it turns out, and you'd have to refer to this paper to see, might be one means by which to provide very acute, very rapid information to these contact events and these spatial details that drive these kinds of motor behaviors. I'm showing you the world's fastest knitter, but of course this also applies to a less fast knitter as well. Okay, so that's just to say that there's a lot of complexity going on in the periphery with respect to the kinds of information that's being extracted. And I want to take that a bit further and say, where within the context of these kind of studies do we need to move? So one thing we really don't know, because we're limited by the kind of stimulation we can give, is the transfer function of those first order tactile neurons. And we could get at that if in the primate glabrous skin, we could use an optogenetic techniques to stimulate the individual endings of that dendritic-like arborization. And in that situation, this is a cartoon of one of those neurons. Imagine you could stimulate one branch and see how that neuron spikes, stimulate another branch and see how it spikes, and then stimulate them simultaneously at different intensities, at different temporal offsets, and eventually stimulate all of those endings. You could actually build up the full transfer function of that neuron and know whether or not what it can compute, what kind of things it can actually extract, and more importantly, whether there's any general principles of what those neurons extract as a population in the periphery. The other thing we need to do is move towards a population level understanding of what's going on. So here, what I'm showing you is example of how this information could be structured at the population level. And that is to take advantage of synchrony across the neuron. So if each neuron temporally structures its responses, you might find that different edges cause synchrony in different sets of neurons. So in this particular example, the 22.5 edge hits this blue, purple, and red neuron at the same time. And if a second order neuron in the cuneate or in the spinal cord was looking for this synchrony, it could respond to that very effectively. So you would turn synchrony code at the first order neurons into a place code at the order of the second order neurons. And a different orientation would then would then target a different subset of neurons, cause synchrony in them, and thus activate a different second order neuron. We don't know if this happens, but in this kind of conceptualization of dealing with what the peripheral apparatus has given us, it's important to demonstrate that this information that we see in the periphery is actually transmitted further on into the central nervous system. Okay, so now I want to back out a little bit and talk about where we are in the periphery relative to the central nervous system. And so this is just a schematic of the kinds of tools that we need to tackle the kinds of questions I've just described for you. And I think absolutely critical one is that almost all the efforts with respect to the peripheral nervous system have been recording one neuron at a time. We need approaches that can deal with the population. And there are now nerve recording techniques or implants in the dorsal root ganglion that might record from dozens of neurons, but we're nowhere near the level that is available in the central nervous system in terms of simultaneous neural recordings. Similarly, fantastic work by Sliman Benzmea and Lee Miller in Chicago have been showing that you can implant Utah arrays into the cuneate nucleus of an awake behaving primate, and again, recording from tens or maybe several tens of neurons in this structure. Again, fantastic, groundbreaking work that needs to be further refined to understand what the true population level activation looks like. Similar for the spinal cord. I want to just say at the end, I think what's critical to all of these affairs is to gain genetic access to these peripheral neurons and receptors. And there's been absolutely groundbreaking work by various people. This particular paper, for example, by David Ginty, but also Alan Lumpkin, many others, to really break down via genetic means what's truly going on in the periphery in terms of those detailed anatomical differences, detailed computational differences, and how they are dealt with across that population of neurons. We've never had this kind of access before. We certainly don't have it yet in primates, but this really opens up completely new kinds of questions, asking what is the distribution of labor between the peripheral nervous system and the central nervous system. And I just end here and say that what I tried to show you today, at least in the context of one specific example, in terms of a particular class of mechanoreceptors in the skin, it also applies for example, muscle spindles and for other classes of cutaneous receptors, joint receptors, and so on, that there's a lot of low-hanging fruit with respect to tackling the function and the detailed action of those receptors using or leveraging the modern toolkit of neuroscience that has been developed mostly for the cerebral cortex, frankly. Thanks very much.
Thanks, Andrew. So let's start. We have a question from the Zoom call from Ilya. So Ilya, you wanna ask?
Yes, thank you, Andrew. Great talk. So you talked quite a bit about synchrony, right? And synchronous activation, synchronous signals, and so on. And so, but it always synchrony between or among spikes. Do you envision that this synchrony, where an absence of spike is a signal, could at some point be important, or do you envision a situation where synchrony between sequences of spikes on individual neurons rather than individual spikes could be important, or is it always individual spikes?
I don't think it has to always be individual spikes. I think it's easiest to think about it as synchrony across set populations of neurons. So depending how you think about that, an absence of a spike can be very, very meaningful to that upstream neuron. But I guess it depends on how you formulate the question. I do think it's very interesting to think about sequences of spikes, which I think could have a very interesting effect depending on those details of the integration time constants and the recurrences within those upstream networks. But for the purposes of this talk, we're just giving the simplest version where you could propagate that information that you get by temporally structuring the responses in the periphery by that relatively tight temporal synchrony. But we don't know. I mean, I think that's a really interesting question. Is we often talk about things like we know that there's a rate code going on here, or some kind of temporal code, or a synchrony code, or a place code. But one thing that I think is really clear once you start digging into it is there's very few examples in the literature where we know what the underlying neural code actually is.
Great. Thanks, Andrew. So we have a question from the YouTube chat from Rachel Seidler. and says, great talk, Andrew. Is there evidence of first order neuron processing and other sensory systems, for example, vestibular, visual, et cetera? And do we give too much credit, do you think, to the brain for processing inputs and controlling outputs?
To the second question, yes, for sure. But I would say for the first question, it's quite interesting. So yes, if you look in detail, for example, in the retina, you start to see the same thing, that there's definitely computations going on in the retina that are that integrate information at a level that was sort of traditionally allotted to the visual cortex. But this gets very tricky. Ultimately determining that the information is there is one thing, but and this is true for the work that I presented too, but ultimately showing that this complex information is used, it's a different kind of challenge. And that's where these kind of causal techniques where you can interfere with optogenetics or genetic approaches to eliminate specific inputs from specific neurons at specific times become critical to disambiguating that question.
Great. So we have a question from Montreal Voss on how do you propose measuring the behavioral outcome of those stimulated first order neurons?
Yeah, this is a very good question. So the problem I see with this, I don't have an answer for it, but the problem I have with this is that I believe, which I didn't get a chance to talk about, that there's a fundamental difference between how these inputs from the periphery are used when they're used for things like conscious reports or percepts, as opposed to online hand control, right? So I tried to allude to that here, but if you're using these inputs to guide your hand, but you try to probe the system, for example, by asking people what they feel, you might completely mislead yourself in the context that you're interested in. I'm not saying that perceptual reports aren't interesting, that those are valid questions to ask. But for me, as somebody who's interested in hand control, I feel you really need to ask them in the context of object, in terms of manual interactions with things, with objects. But now how to precisely gear that up with optogenetic stimulation of the first order tactile neurons, that's of course a large challenge, especially because this needs to be very spatially specific stimulation. The one good thing we have going for us that makes it at least hopeful is at least for the first order tactile neurons, you don't have any efferent innervation. So in that sense, you can at least hope that the things that you do passively might transfer to the active case in terms of that very local distal processing. But you have skin mechanics you need to consider. There's lots of other factors at play. But in terms of that neuron itself, you don't have any efferent innervation to it.
And from Val Gritsenko, how do you see the role of skin receptors in continuous versus intermittent control of reaching and grasping?
Yeah, this is a fantastic question because we just basically wrote a grant about this. So I think that this, I really, and this is not my original idea by any means, lots of people have emphasized this before, but I really think that these contact events are where the system wakes up for short periods of time. So the actual reach, when you develop that reach, the real salient information comes in at the endpoints of that reach. You get those contact events, and from there, you really update the system. And one thing we're very interested in understanding in our lab in collaboration with Jorn Diedrichsen, for example, is how those contact events help the system to propagate through sequences of motor actions. So as you touch something, you're going to have to, depending exactly how you touch it, you're going to now have to slightly update the next motor plan because you didn't grasp it exactly the way you wanted to. And we're really trying to understand how that process plays out. So that makes very specific predictions about how these cutaneous inputs propagate through to for example, the primary motor cortex or premotor cortices, where these transitions might be occurring.
So there's a bunch of other really great questions, but unfortunately, I think we're going to have to move on. But thanks again, Andrew. And now our next speaker will be Amy Bastian, and she's coming to us from the Kennedy Krieger Institute, where she's the Chief Science Officer, and she's also a professor at Johns Hopkins. And her work really is a fantastic work at the interface between neuroscience and robotics and rehabilitative medicine. And really, she's trying to understand the neural control of human movements, and she'll probably say it more elegantly than I can. So Amy, go for it.
All right, I'm unmuted. Thank you. Thank you for inviting me. This has already been great fun. I'm going to take a little bit of a different approach and tell you about what I would really like to have. And this is sort of a 30,000-foot view. I study human behavior, and I'm very interested in how human behavior develops, how people learn, and how people deal with injury. And so today, what I'm going to show you is that I think we need something that I'll call growth curves for the development of human behavior. And I'm going to walk you through this in an example and explain why this would be important, how it would be useful, not necessarily how we'll do it, but the idea of having a really granular look at how people, humans, and other animals develop motor skills, motor learning abilities, and sensory processing abilities. So an example I'll give you today is about motor learning mechanisms. And I'll just show you a little bit of the data that suggests that things don't necessarily emerge when you think they might. And they certainly don't reach maturity when you think they might, based on our clinical expectations. And by knowing that, we will be able to study the mechanisms and interactions of learning in much more of a de novo state. So in development, you don't have a lifetime of experience. And so I think it's actually a terrific system to be able to start to understand circuits operating at the beginning. It'll allow us to interpret structural studies of brain development, which are very popular for understanding pathology, identify deficits early, and determine what mechanisms are available to be leveraged for treatment. So many, but not all of you probably know a lot about human brain development. So I'm going to give you a few quick facts. This is actually back from the late 1950s. This is sort of a landmark study. When you're born, your neurons are still migrating. And when you get to about, during infancy, you're developing about 700 synapses per second. And so the brain is very active and changing rapidly. A 2-year-old has twice the number of synapses as an adult, and brain volume increases 4-fold over these first years of life. So there's a lot of action happening, and we don't understand how things are coming online during this time. Importantly, the brain volume then decreases, and so here are 4- and 6-year examples of a cortical, a section through cortex. Things are pruned, and connections that are used are really the ones that are more likely to stick. So in this context, we really don't have much good information. We have developmental milestones, but we don't have much good information, granular information, about how different systems come online for learning and controlling and sensing. Now, I use the word growth curve, and everybody's probably familiar with growth curves. This is an example of what you would see at the doctor. And I'm going to zoom in on head circumference. So head circumference has been a really useful measurement, even though it's a very crude measurement. It allows you to understand what percentile your infant is in as they develop, which gives you a proxy for something about whether their brain is developing appropriately or not. Now, what I'm looking for is something that extends way beyond this, but still follows this general concept. So this is not unique, by the way. Large-scale MRI studies are doing this very thing for structural anatomic changes in the brain. This is an animation from a PNAS paper from the NIH many years ago, showing that maturity, which is indicated in blue when the cortex has shrunk a little bit, starts in the primary sensory motor cortices and then fans out. And so you can see this in the animation. This is sort of a developmental time course of the thickening and then shrinking of the cortical thickness. Now, there are many different studies right now doing this, looking at different anatomical markers of brain development, but I think that they're missing something. And this is a detailed time course of the different behavioral abilities coming online during human development. So there are smatterings of things out there that describe this, but there is no good systematic look at it. Now, what I'll do now is take you through just an example set of data, just a couple of slides. And in these slides, I'm just going to show you how it can be counterintuitive, or you may not expect development to take quite as long as it does to allow for normal control of, in this case, human walking and learning of human walking. So the study that we use is we've used for many years and is now being used in rodent model with Megan Carey, who's not here today. We use a split belt treadmill. We ask people to walk on the treadmill and we track their motion. We record the position and the joints of the body. Sometimes we record EMG patterns. We measure the step sizes as they progress. And then we do an experiment where we, unbeknownst to them, split the belts, and then they feel it, and then they have to deal with it. And then we tie the belts. We make the belts move at the same speed. So in the next slide, what I'll show you is how a human actually deals with this. This is an adult human, and then I'm going to show you kid data. So this is an animation of a balloon guy, and he's walking in the tied belt condition. And you can see the legs move in antiphase, and the step sizes are even. That's the distance between the steps as he walks. The next individual, or the next balloon guy, shows what happens when you split the treadmill belts. People limp, and they limp in a very specific way. They take a small step and then a big step. They still move in antiphase, so that's locked in there. But the yellow leg starts to walk a little bit in front of the blue leg, as you can see right here, and the legs aren't in perfect antiphase. Now, the beautiful thing about this particular paradigm is you don't have to think about this at all. The nervous system, through adaptation, changes this pattern to what I'm showing you here. This is late adaptation, and the legs reestablish antiphase, and they can move in a nice, even stepping pattern. When you remove the perturbation, what you see is a phenomenon that we call an after effect, where they limp in the opposite direction. And this is shown here. This is tied belt walking again, but now we can see them expressing this motor memory that they've formed. And we don't know if this unlearning of the perturbation is really truly a learning process, or if it's a switching process, which is one of the things that would be very helpful to know. Okay, so that's the basic paradigm. One of the things that surprised me was that this learning has sort of a protracted development in childhood. And so what I'm showing you here is a learning curve for learning the step symmetry pattern. Zero step asymmetry is normal. When you perturb an adult, they produce this nice double exponential pattern. And then when you return to the tied belts, they unlearn it. And this occurs over about 500 steps. Now, if you study loads of children on the treadmill, which we've done many times, you find that there's a developmental trajectory of this learning. And so here what I'm showing you is the youngest group. This is a lot of inter-subject variability. They learn in a more linear fashion. So do the 6 to 8-year-olds, the 9 to 11-year-olds. And then as you start to reach 12 to 14 and 15 to 17, you can see that you get a little bit more of an adult-like pattern where they start to learn in a double exponential manner. Now, so we were surprised that by 9, kids were not learning these walking patterns normally at all. They were slower and they seemed to lose some features of the walking. Now, on the next slide, I'm going to show you another phenomenon, which I think is really interesting, and again, points to the fact that we don't understand how these systems come online. So in this set of experiments, we did the same thing. We took children and we measured their savings. And what savings is faster relearning after you, quote, wash out the memory. And so here, what I'm showing you down below is the data from a 15 to 17-year-old group. Exposure 1 is the light purple. And so they learn the treadmill perturbation. We send them home. They come back the next day. and we totally wash them out. So then they start from here, this dot. And then we split the belts again and re-perturb them. Now, this purple shows a nice savings effect, whereas so does the blue, and these are in 12 to 14 and 15 to 17-year-old kids. But you can see here that our 2 to 5-year-olds, 6 to 8-year-olds, and 9 to 11-year-olds act as if they have never seen this perturbation before. We wash them out and they learn in exactly the same way. And this is not a problem with retention. So if you do the same thing in children and you adapt them, so we'll take, for example, this green curve, you send them home, you bring them back the next day, and you put them right back on the treadmill, they can retrieve that context-specific memory. So it's really the washing out and relearning the memory or re-engaging the memory that's problematic. The last thing I'll say, I know I'm close to overtime, is that these processes, understanding them, actually let us do things with patients that we can't necessarily do easily in rehabilitation. So this is a child who had a hemispherectomy, and we know that this learning process depends in part on the cerebellum. And so this child, if I ask her to voluntarily control her walking, she has a great deal of trouble doing it. If I ask her to voluntarily change it and improve it, it's hard for her to engage. And so here is her walking pattern at baseline. You'll notice she comes up on her toe and she takes asymmetric steps. She has trouble swinging that leg through. Passive dynamics are not working for her right now. But they can. And so what we do is we expose her to this split belt learning process. And we actually make her limp worse. And we do this by making the leg closest to us go fast. And you can see she's even limping more. But this drives, we think, these perhaps subcortical networks to learn a new pattern. And then if you go back to the tied belts and compare it to what you saw at baseline, now she does not come up on her toe and she can take nice even steps. So what we've done through this learning process is allowed her to achieve a pattern that she can voluntarily. So I'm gonna wrap up by saying that I would like to understand what this child is gonna be able to do. I'm not gonna try to understand why this child's parents put a pacifier in his mouth and strapped him to a snowboard, because I think that that's an entirely different kind of question. But I'd like to know, what could I expect this kid to do? Would he be able to, what would he need to be able to learn how to snowboard? Does he have the right sensory functions online? Can he integrate vestibular, proprioceptive, and visual information in order to be able to deal with this? Can he control the redundancy of his movement? And how does this change over development? It's going to take some computational power to understand the interactions in these processes. And we also need to understand how it relates to detailed mapping of brain structure, if that structure is going to be useful to us in order to understand how humans and other animals behave. So I know I came from a human-centric point of view, but you knew that coming in. And this is just a slide to thank the people who do this work and people who fund this work. Thank you.
Great. Thanks, Amy. So let's start. I have a question from Flip. Do you want to take it away? You're muted.
Hey, Amy, thanks. That was really great. So you showed that the learning curves, and in particular retention, is quite different during development, way late into development. Do you have any thoughts about the anatomical basis for those differences?
Right. So there's a lack of savings, but there is retention of a memory. And so it seems that they're able to switch contexts. In other words, they're able to say, I have this motor memory. It's for this crazy treadmill this lady put me on. I'll go home. I won't use that memory. I'll come back and I'll re-engage that memory. When you wash it out, I mean, one model is that it's a cerebellar cortical site where it can be washed out until it eventually goes to the nuclei. This is the old work from Javier Medina and Mike Mauk. It's possible that this could be in the brainstem. I don't think I'd put it in the cortex at this point, although cortical information is very important for the context, for any animal to understand context. I will tell you that children also generalize more than adults do between contexts. So that to me means that maybe they don't have as ideal cortical development for establishing they're in a specific context. So that was a long answer of saying I don't know for sure, but at least it's a few sites that are possible.
Thanks.
Other Amy, do you have a question? Which one will be other Amy will flip later in the workshop?
Right, I'll be other Amy later.
Yeah. So really great talk. I was curious because the map you showed of the cortical development suggested that actually motor areas consolidate earliest.
Mm-hmm.
And yet it seems like there's a really big lag between the cortical development that you're seeing and these learning mechanisms coming online. And so I was curious if you had any ideas about where that lag comes from.
So I sort of showed you one example of development of the brain. I showed you cortical thickness. What I didn't show you is myelination pattern. And so that takes a little bit longer to occur. I didn't show you, they constantly lop off the cerebellum, which is really annoying. So cerebellar development is actually quite protracted. It doesn't reach its peak volume until around age 12. And then it starts to refine. And so I think that the kind of learning I'm studying here is really, it's got a lot of subcortical components to it. It's not all spinal, we know that, because we can get rid of it when we lesion the cerebellum. So my thought is that it's the cerebellar development that may be limiting it. And of course there are some longer term changes that occur with myelination in the corticospinal system.
Thanks. Abby, did you have a question?
Yeah, Amy, that was super fascinating. I was interested in the child that you were looking at with cerebellar damage, who had a limp, and then you exaggerated the limp.
Cerebral damage.
Oh, it was cerebral. Oh, I apologize. I missed that. Oh, I see now. That's a huge cerebral damage. Well, regardless. So do you imagine that this child had a sort of a different motor goal that was tolerated in the sort of pre-adaptation condition, and then that there is just a different set point that's available, or how are you thinking about what is being achieved by this system? What's the system driving at?
So you could argue, so A, we don't know, but B, what we do know is that these are kids that have seizure disorders that affect one hemisphere, and they're having seizures all the time. This is about 3 weeks after this child had her hemispherectomy. And so it's likely that the nervous system was reorganizing and shifting control before the hemispherectomy. If you did this to an adult, it would be a very different ballgame. But she also had a sick hemisphere. And so it's likely that control shifted to some extent, that the cerebellum still could be engaged. And whether she has a different set point or not is hard for me to know. She certainly behaves in a way that doesn't engage her full capacity. And so one of the things that we're interested in understanding is if her sensory perception of her limbs is one of the things that's limiting this or changing this. And we're doing that work right now actually in adults. So perception of perception, sensory information reaches the cerebellum still of this kid, but the perception of her limb is what's not intact. And so that's another piece of this.
We have time for one more question. I know there's a bunch of great questions on the Zoom call, but I'd like to ask one from the YouTube channel, which is from John Jacob, who asks, it seems that the young children are not using error correction to learn. Is it more like babbling?
Oh, no, I would argue John, it's a great question. Really young children, I think, probably use reinforcement mechanisms more with exploration and reinforcement. That's my hypothesis. The really young kids, they don't go in the wrong direction. So I guess it could be possible, but we do seem to see them respond to errors in the way that we see adults respond to errors. It's just they lose that initial sharp part of the exponential. And so we're working on this right now to try to understand it. We need you animal model people to interrogate the brain in a way that I can't. And again, if we had behavioral data that we could compare across lower animals and humans, that would go a long way in me being able to really say something about neural substrate and mechanism. So that's why I'm excited about this group and being asked to speak. So thank you.
Well, thanks a lot, Amy. That was really fantastic. And I always like it when you have a talk that ends on a note of measuring more behavior. But so our last speaker for the first part of our symposium is Chethan Pandarinath, my colleague over normally a couple buildings over at Emory in the biomedical engineering department. And Chethan does work really on both really fantastic experimental and analytical / computational work, thinking about motor control and how to analyze it and how neurons encode behavior. So Chethan, and also it looks like you need to flip your screens.
Huh. Thanks,
just do the
Gordon.
swap. Just do the swap screen on your.
Appreciate it. Yep. Okay, can everybody hear me and see slides now? All right, awesome. Thanks very much. My name is Chethan Pandarinath. I'm an assistant professor in biomedical engineering and neurosurgery here at Emory and Georgia Tech. Thanks very much. This is a really wonderful group to be a part of. Quick recognition that obviously the last few weeks have been especially hard for people of color in academia, and I think the week has added significant stresses for immigrants also. So let's just start off with the hope that everybody's watching out for their friends and colleagues that might be impacted. Okay, a 10-minute talk on the future of motor neuroscience, prepared in the middle of a global pandemic. So I think Gordon appropriately described this as a patently ridiculous assignment. My main regret is that some of this kind of represents a general point of view, and I don't have time to point out all the great related work in the field. But anyway, let's give it a go. So brain-machine interfaces are assistive devices to help people who are paralyzed. And some of the many exciting developments in the last few years have been restoring tactile sensation, voluntary limb control, rapid brain control typing, and even synthesis of speech. And these developments are really promising, but another key aspect of BMIs is that they really put our knowledge of motor control to the test. So we start with our assumptions about how the brain controls movements, and we use those principles to drive a BMI. And when we're wrong, the system breaks. So I'll quickly show you a video from 60 Minutes from a few years ago. This episode highlighted some amazing work by Jen Collinger, Andy Schwartz, and colleagues at Pittsburgh. And a really admirable point here is that they were upfront in highlighting some of the challenges they were facing. So there are obviously many different challenges facing BMIs. Let's just focus on one particular aspect.
Andy Schwartz believes that will help with some of the things that Jen has trouble with. Okay. For example, sometimes when she looks right at an object, she can't grab it.
Okay, I'm gonna take the cone away. Just go ahead and close it. Oh, sure. As soon as I take the cone away, there's no problem. But as soon as I put the cone there, she can't do it.
Why is still a mystery.
Okay, so what did we see there? The participant could control grasping with the robotic arm when there was no object, but when an object was placed in front of her, she could no longer control her grasp. And they learned a relationship between motor cortical activity and grasping behavior in one context, but that relationship didn't generalize. And they later introduced solutions to this specific problem. But I love this example because it highlights a key challenge that we face both in brain-machine interfaces and in studying motor control in general. And at the core of this issue is the fact that relationships between neural activity and behavior learned in one context may not generalize to new contexts. So, I think part of this stems from the fact that our fundamental approach here hasn't really changed since the seminal studies many decades ago. We typically try to constrain behavior as much as possible to isolate key movement dimensions. And then we make repeated measurements of stereotype movements. And then typically we come back the next day and do the same thing again and again. And eventually we repeat that with the next subject. So let's imagine that we want to apply that same approach to characterizing reaching and grasping behavior. So we could start by setting up the reaching part, maybe 8 targets in a workspace. Because neural activity is highly variable, we probably need to make each reach multiple times, let's say 20 times. So that's about 160 trials. Okay, but what if reach encoding changes when we're grasping an object? Well, maybe let's do the same thing, but let's pick up an object in the center on the way out to the periphery and leave it there. So that's about 320 trials. But maybe the direction we're transporting the object matters. So we've got to do the same thing, but pick it up at the periphery and leave that in the center for 80 trials. Soft versus hard objects, okay, 960. Maybe different grips, different hand orientations, maybe there are occlusions in the workspace. I mean, I think you get the point here, right? But obviously, as the number of behavioral configurations gets higher and higher, we get to the point where these experiments become no longer feasible. So I think we're all kind of supposed to have a quest and a vision here. So my dream here is that we're able to create truly massive data sets. And in these data sets, we'd monitor activity during complex behaviors and over extremely long timescales, maybe over months or even years. And hopefully we'd have these data sets from many, many subjects as well. And I think these massive data sets would allow us to uncover the relation between activity and behavior with much greater specificity than we currently can. Now that of course isn't necessarily a profound idea, but let's talk about how we could actually make sense of those data sets. So my proposal is that we focus on the computations underlying the neural population activity and do that using dynamical systems. And second, to really make sense of data over very long timescales, we can leverage the fact that we see consistent dynamics over those timescales. Also, that single trial analyses really eliminate the need for repeated behaviors. And the last idea here is crazy enough that I'll just leave it for the end so we can conjecture. So what do I mean when I refer to dynamical systems? So really briefly, the idea here is that the activity of all the individual neurons we observe in a brain area has some shared structure, which reflects the fact, of course, that neurons are part of a coordinated network. And while the activity of individual neurons may be quite confusing, activity of the network can be readily understood by investigating the dynamics underlying the neural population. So I'm going to show you some visualizations in this talk. In each of these, I'm showing the shared structure underlying the population's activity. So we typically call these neural state space plots, where rather than plotting the activity of individual neurons, each axis shown represents a pattern of correlated firing across neurons. So a quick example, let's take this study from the Shenoy lab, which studied cortical activity, motor cortical activity in monkeys making a wide variety of reaching movements. You see that on the top left. They recorded from 200 neurons in motor cortex. Now for each movement at the top left, you see a corresponding trace at the bottom. These represent dominant patterns of activity in the neural population just before the monkey starts moving. The key takeaway here is regardless of the specific movement the monkey made, there's clear organization and structure in the neural population activity, which we call the dynamics. So this is a really simplified view, but it gives a nice visualization of the consistent structure. Now, using techniques from unsupervised machine learning techniques, we can now pull out these patterns from neural activity, but now on individual trials. So in each plot here, in the earlier plot, each trajectory reflected an average across 20 or 30 movements. Now I'm showing you activity for each individual trial. So 2,100 individual reaches across a 1.5 hour session. So there are different techniques to do this. I'm just focusing on what we do in my lab. But think about it this way. Rather than using many repeated trials to denoise data and find consistent structure, we use the knowledge that the population activity reflects an underlying dynamical system with rules that determine how activity evolves over time. And by modeling that system, we can reject kind of patterns that aren't consistent with the learned dynamics, which we can treat as noise. But what we find is that when we do this, we actually uncover representations that are substantially more informative about behavior than previous approaches. So it tells us that there's something kind of veridical about these dynamics that we're pulling out. So how does all that relate to the massive data sets I want so badly? So first, let me show you that leveraging dynamics allows us to leverage data over very long timescales. And so a key challenge with data over long timescales is just that chronic recordings using technologies we have right now are unstable. You know, we have neurons that are coming in and out constantly. It's very hard to keep stable recordings over substantial time periods. And this has been a problem, you know, both in brain-machine interfaces and in neuroscience in general. But as multiple groups have shown, it seems like these underlying dynamics are consistent across very long timescales. So what I'm showing here is some work led by Dan O'Shea in a previous publication, where we recorded from separate recorded populations spanning 5 months. but we viewed those populations in the context of a single underlying dynamical system. And what we found in that work was that a consistent set of dynamics could describe the data across 5 months. So that's what we're showing here. Each one of these plots is from one of 44 different sessions spanning 5 months, and all the traces are individual trials colored by the monkey's reaching direction. And we find these consistent dynamics across this time period. And what's really important here is that by finding these dynamics, we were able to decode the monkey's behavior more accurately than we could have with any of those individual sessions alone. So this is really a technique to get at really kind of large time scale recordings. So second, I wanted to show you that single trial analyses really eliminate the need for repeated behaviors. And what I'm going to show you there is just completely a proof of concept. So now we're going to apply these dynamical systems methods to data from an unstructured task. This is an amazing data set from Joseph O'Doherty and colleagues from Flip's lab, Flip Sabes' lab. And the key point here is that this is a random target task where the monkey is just kind of chasing targets around a workspace. So this task probes a lot more configurations than others. Movements are different lengths and speeds. They start from many different positions. So this is a non-stereotype behavior with no repeated trials. And we put that kind of into these algorithms without any knowledge about the task, the target onsets, or even that there are trial boundaries. And it hasn't been specialized for the brain area being recorded. So let's ask what the representations pull out by these algorithms look like. So here I'm showing you population activity for about 150 individual trials. And what we're looking at is what the population activity does in each trial just before the monkey makes a movement. And you can see that the system has learned some really stereotyped patterns for what's happening, but with some clear variability across trials. Now we're just going to go back post hoc and color these individual trials by which direction the monkey happened to be moving in. So when we trained the dynamical system, it didn't know anything about the monkey's behavior. It's just trying to describe the data as the output of a dynamical system. But post hoc, we can see that the learned representations are clearly informative about what the monkey was actually doing. What I'm not showing you here are some of the scientific implications. We can replicate previous findings about the structure and organization of preparatory and movement activity using a task that was not at all specialized to try to pull that out. And second, in terms of applications like BMIs, these types of representations substantially increase our ability to decode from the monkey's behavior on fast timescales. So I'm hoping to convince you here that without the highly stereotyped behavior of current paradigms, we can still tap into the computations being performed by the neural populations. So, two ideas for how we could go to longer timescales and how we could eliminate stereotype behaviors. The last thing I want to mention, of course, so remember what I told you about kind of stitching data across multiple sessions from the same subject. We could pull out consistent structure by knowing that there was a consistent underlying dynamical system. So this is complete conjecture, but maybe these same concepts apply when we're trying to pull out consistent structure, but maybe across subjects. So, the hypothesis here is just that the same part of my brain performs similar computations to the same part of your brain. So if we're focusing on the dynamics which are related to the underlying computations performed by our brain area, then maybe we can leverage these new kind of machine learning techniques, especially from areas like domain adaptation, to try to leverage data sets across many subjects and try to build, really, really massive data sets to get at some of these complex and higher dimensional behaviors. So I didn't get to talk about the fact that some of these same concepts hold up at the level of populations of muscles. So that's one of the directions we're really excited about going, especially in this collaboration. There's a completely separate set of questions about what learning looks like, over long timescales and how that interacts with these consistent dynamics. But obviously time. So I hope everybody took that with an appropriate grain of salt, and I'm looking forward to questions. Thank you.
Thanks, Chethan. It looks like Sam has a question.
Hi there. So that was a fantastic talk, Chethan. So what I was thinking about is the following. So, okay, so you make a convincing case that we don't necessarily need to stick to the sort of the traditional paradigm, which I've definitely done a lot of work on, during my grad days, when you have somebody make 1,000 reaches, all from the same point, start point to, one of 8 targets. But one thing that's really good about that is that it gives you a really natural way to look at generalization. So, you overtrain somebody in one part of the workspace, and then you move them, whatever, 6 centimeters over and look at how it generalizes. So I guess it's a pretty open-ended question, but how do you think about generalization, both from the perspective of what the brain's doing, because, when we actually make naturalistic movements, we don't, do the same movement 1,000 times, but also from the perspective of the, the machine learning algorithms that you want to apply. It seems like generalization is the, one of the huge strengths of machine learning, but also one of the, maybe one of the challenges if you really want us to, all throw away having animals do, identical or similar trials.
Yeah, it's a really interesting question and a great point. And I think I think that relates to the idea of trying to avoid actually stereotyped behaviors. It may be a good idea to try to avoid stereotyped behaviors just to try to get greater generalization performance. So, I mean, I think a common saying in machine learning is that, things like deep neural networks are really great at interpolation, but they're not always great at extrapolation, right? So you want to make sure the data sets that you use to train your algorithms and uncover representations as best as you can span the set of behaviors that you want. So I think you'd actually expect, like, if you started with a really constrained behavior and overtrained, you sort of overfit your system to that condition. Whereas if you are able to use like less structured data that spans a lot of behavioral conditions, you'd probably expect much greater generalization performance.
Okay, thanks.
So Rui, do you have a question?
Yes. Yes, Chethan, hi. Great talk. So I wonder if you could elaborate a bit about computationally how your methods, especially you mentioned in the last slide, you could apply the same thing for large data sets on muscles or even single units on muscles. And is there a way you conceive establishing a computational framework that would get us at the relation between the dynamics of the brain activity, let's say in the cortex, and the dynamics of the muscle activity? Some algorithmic rule, like between the dynamics, the different dynamics.
Thanks, Rui. I think that's a really it's a really interesting question. How could we potentially use these techniques to try to understand communication between different systems that we think maybe have their own intrinsic dynamics? So, for example, at the level of the muscles, there's probably a lot of intrinsic dynamics in the spinal interneuron circuitry itself, which of course is receiving descending commands from different brain areas. And I think if I'm just going to conjecture here, what we'd probably like to do is separate out what we consider dynamics, which are things that are predictable based on the current state of the population, from inputs to the system, which aren't predictable based on the current state of the population. So, basically to try to understand maybe what a command looks like, can we say there wasn't information in the population to predict what happens in the future? So that reflects the fact that an input must have come in to perturb the system in some way. So things that are predictable are likely to reflect intrinsic dynamics, and things that are not predictable are likely to reflect inputs potentially from different brain areas or just populations that we don't happen to be recording from.
So we have a question from the YouTube chat from John Krakauer. I can't quite do this in his voice, so I'll just have to say it in mine. Can you say more why you call the trajectory as providing insight to the computation rather than the derived consistent object?
Okay, providing insight to the computation rather than, I'm not sure, I didn't quite understand what the derived consistent object meant, but there's when I say insight into the computation, there's a lot that I'm not unpacking there. So I think one of the key things that's been studied when we focus on motor cortical activity, especially over the last few years, is understanding how the patterns of activity we see in motor cortex relate to the process of preparing and then executing a movement. So for example, there's been some great work showing that patterns that seem to be related to preparation or at least pre-movement activity that seems to be indicative of what the upcoming reach is are often orthogonal in a neural population state space from patterns that seem to be related to execution of the activity or the moment by moment details of the activity. And so what I meant to say is these types of techniques help us tap in, if we kind of believe that this is part of the computational role of motor cortex, and that these the organization of the activities related to that computation, the ability to prepare without eliciting a movement, those sorts of things, and perhaps that preparation, there have been studies that show that it may be related to learning, as you well know. So perhaps, these types of techniques that try to kind of model these dynamics and let us probe the relationship between activity in these different state spaces can help give us insight into computation, is all I meant by that.
Leah, do you have a question?
Yeah,
and be quick. You have 1 minute.
Okay. So Chethan, thank you for a great talk. So a couple of related things, right? So you have you're emphasizing this low-dimensional dynamical systems in specific tasks. To which extent these dynamical systems actually are generalized across tasks, and to which extent the overall dynamic actually remains low-dimensional? Is it even meaningful to talk about low-dimensional things? Or are we throwing the baby with the bathwater?
Sure, yeah, and I didn't mean to say that these techniques are only good for modeling low-dimensional dynamical systems. I think we can still gain insight into higher dimensional systems, but we may be limited just by experimental considerations there, like potentially the number of neurons we're recording from. But I think kind of related to your questions, there's been great, for example, theoretical work by Surya Ganguly's lab that suggests that a lot of kind of the low dimensional structure that we see is related to kind of like the stereotype tasks that we tend to use. And I honestly think it's just an untested question. As we're able to move to higher degrees of freedom behavior or higher dimensional behavior, will we see consistent dynamics? Do we need more complex dynamical systems? It's likely that we do, to kind of describe those higher degree of freedom behaviors. But to be honest, and this is where the conjecture comes in, what I'm trying to motivate is that that's where we need to go. We need to start building those data sets to be able to ask those questions. And I don't think we can with the current kind of way that we're probing the system.
Thank you. Thank you.
Right. One last question, and this comes from Jerry Fishbach. Have you noticed any difference in the dynamical system as a function of age or sex?
Well, it's a really wonderful question. I think there's a lot of that. I think it's similar, both kind of changes related to age and sex, and probably also changes related to learning are kind of very interesting points. I think as far as I know, we don't have that data right now to be able to evaluate kind of over really long time scales. Do we see fundamental changes? I think there's some great work by folks like Byron Yu and Steve Chase and Aaron Batista that have looked at how manifolds and dynamics change with relation to learning, which is something we can kind of prompt over shorter time scales. But again, I think this is sort of where I'd go to investigate that is if we really have these long time scale data sets, then we start with the assumption that dynamics are consistent, and that gives us some set of representations. And we can almost potentially use that as a prior to say, all right, now that we have this newer data, recorded from many months or years out, what aspects of it are different than the consistent dynamics that we assumed? So, again, conjecture, but I think this really motivates being able to start to collect these types of data sets to ask exactly those types of questions.
All right, thanks. So I can see all sorts of great questions on the chat, but unfortunately, I think we're already way over. So, we'll come back, and let's say we had a 10-minute break scheduled, so let's just plan that. So we'll come back at 11:50 a.m. Eastern time sharp. And that we'll have Flip Sabes will be the talk right after. But before we go, I just want to make a quick advertisement for the next Theory and Modeling of Living Systems at Emory workshop, and that will be on July 31st, and that will be on the interface between soft condensed matter physics and biophysics. That'll be what can soft matter physics teach us about biological function, with what being in parentheses. That will be organized by Jennifer Reeser and Daniel Sussman, both from Emory Physics. And please follow Emory TMLS either on Twitter or Facebook for further announcements on that. So I will see everybody at 11:50 Eastern time, and we'll be back.
Okay, welcome back, everyone. Excuse me. All right, so thank you for sticking around. I hope everyone had a good, very quick lunch, midnight snack, depending on your time zone. So I'm very happy to be introducing Flip Sabes as the next speaker in our symposium. Flip is a professor emeritus at UCSF. He's also one of the founders and currently a scientific advisor at Neuralink. Flip asked me to mention that he's not representing Neuralink in the talk that he's going to be presenting today. So Flip has a long history of doing really excellent experimental, theoretical, and engineering work on motor control in both humans and non-human primates. Flip also, I should mention, happens to be my former PhD advisor, and it's a long-standing dream of mine to moderate a talk that he gives. So I really want you to bombard him with challenging questions, and please don't hold back. So with that, over to you.
Okay. Hopefully you can hear me and see my screen. Can I get a thumbs up if that's true? Great, excellent. Okay, so thanks, Sam. That was an overly generous introduction. So before I start talking about tools, I think it's good to just ask the big picture question here. And this is going to be kind of a big picture kind of talk. And I think the key question that addresses the field is, how do we get so good? How does motor control get so good? And by good here, what I mean is the ability to flexibly process sensory signals, information about the world, knowledge of one's goals, etc., in order to respond in an appropriate or even optimal way across a wide range of contexts, and also the ability to robustly respond to changes in those signals. So we want to understand what, in this talk, I'm going to perhaps goofily refer to as the general intelligence of motor control. Okay, so the first question is, if you wanted to study that, where would you look? Which brain area should you record in? You know, when I was a graduate student, it was common for researchers, or even a whole lab, to spend a decade studying essentially one task in one brain area. But of course, even simple movements like reaching to a point of light or making a saccade, which were the staples of motor control in that era, even those simple movements require a coordination of a large circuit that spans the brain, spans the nervous system. And so obviously we need to, that's not going to work. We need to study circuits, not areas. But which circuits? What does the motor circuit look like that underlies general motor intelligence? Well, here's a picture of the motor circuit from the perspective of someone who studies muscle control from cortex. Here's a picture of the motor circuit from the perspective of someone who studies somatosensory feedback. Here's a more ambitious, maybe, picture of the motor circuit from the perspective of sensory and cognitive control of movement. And here's a schematic circuit of the motor system from the perspective of learning, and I'll come back to this one later. So the point here is that understanding general motor intelligence requires studying the system from all angles, and not only that, studying the whole system. So in the dream experiment, we'd record from all of this. All right, well, dream experiments aren't really that interesting if they're not even close to possible. So what tools might we use to get there? Well, optical techniques have gotten pretty impressive, especially in rodent, where we can use genetic tools. Here's an example of a recording from over 1,000 neurons across most of dorsal cortex in a mouse. Thin film arrays are also making significant progress. Here's a picture of an active array that was used to record from over 1,000 electrodes on the surface, or ECOG, across a big swath of somatosensory and motor cortex in macaque. And of course, we saw a similar device at the beginning of the session from Sam and Gordon, used to record EEG. There's also been progress in the development of thin film arrays for penetrating electrodes, either thin film or thin silicon electrodes. On the top, on the left there, is an illustration of the sewing machine approach that Tim Hansen, Michelle Maharbis, and I started in my lab some years ago. And in fact, a big part of the motivation for this approach was the idea that if you had these kind of flexible electrodes, you can target thousands of sites across many brain areas, say hundreds of neurons and maybe say up to 10 brain areas. The figure on the bottom and the left there shows some of how this approach has been developed at Neuralink. Here, this is a published image of 3,000 electrodes that were placed across the dorsal surface of rat cortex. Okay. Now, while these techniques are all progressing fairly quickly, I have to admit that it's pretty hard to see how any of them would be able to access the full motor circuit anytime soon in a given animal. But that doesn't mean that we can't study the full circuit. So we can, of course, use a combination of approaches, and we can do that across multiple animals. So, I don't think you can study one brain area at a time, because if you do that, you only have trial averages and you lose information about how information flows from area to area across trials from moment to moment across the circuit. But you don't really have to record the entire circuit at the same time. You can tile that circuit in sort of sub-functional units, and if you tile it well enough, you can get a picture of the entire motor system. But still, that's probably a bit much to ask from a single lab. So how could we do that? Well, one approach is the sort of brain observatory idea, but and I think those are good ideas. But there's a big energy barrier to getting those set up, and how many of them can there be anyway? So here's another idea. You could do this across multiple labs, and here my inspiration is the really brilliantly conceived International Brain Lab, which is a collection of about 20 labs, both experimental and theoretical, that are studying different aspects of a single decision-making task in mouse. So I think something like this could be brought to bear in more areas, and in particular in motor control. And then I will just say one more thing, which is that I think ultimately, if the goal is to understand human motor control, not just biological motor control, which for me it is, then I think the best approach is going to be to use multiple species, in particular, probably a combination of mouse and monkey. I don't think that that's a controversial claim, but if it is, I'm happy to discuss it more in the question period. Okay, so the question that we were asked is, what tools are we missing? And I guess my answer to that is, maybe the tools are getting closer than we might have thought to do even really ambitious things. And maybe what the harder part is rethinking how we do science. Okay. So that's good. Now, if we could do that, if we could record from the whole circuit, what would we do with that? What questions could we ask? And again, I'll go back to the beginning. How does motor control get so good? So what do I mean by that? And here I'm going to step outside a little bit of my comfort zone, and I'm going to take up Amy's call to the animal model people. And I promise this was not actually planned in advance. That to record longitudinally, I propose, in animals during the course of motor development. Now, Amy showed earlier that most of what we know about motor development is known from anatomy and behavior. But I think that if we want to understand how general motor intelligence arises, that's not enough. We also need to include, if we want to get to the computational principles here, we need to include circuit-wide physiology. So I'll give an example shortly, but before I do, I want to reiterate a point that Amy made earlier, which is that development is actually a lot slower than you might think. And I think from the perspective of this proposal, this is a good thing, because it gives you a lot of time, lots of data points can be collected at different stages of development. So just for example, in humans, as Amy's already shown us, it takes years to decades to fully develop. One nice example I saw when I was looking up some papers for this talk is the link between the grip force that you apply on an object and the load force that you experience as you move it up and down, for example, is tightly connected. But it turns out that tight connection doesn't even come into play. It doesn't come online until about 10 years of age, which I thought was surprising. In macaque, the time scale of development is months to years. So for example, it takes 3 to 6 months to develop a precision grip, and the cortical spinal tract isn't even finished developing until almost a year. And then in mouse, the time scale is somewhere on the order of days to weeks. So there's an opportunity here, I think, to do the sorts of experiments that I have in mind. So why? What can we get from this? And I think to answer that, I want to go to the analogy of machine learning. Deep learning, machine learning has come very far. Obviously, it's of wide interest in the entire world and in our community. And although the final network that you get from these techniques is certainly interesting and useful, all of the work really it's not the network. The work is all in data parsing, in learning rules, in the architectures that you choose, in the constraints on the learning rules, etc. That's where that's where the meat is. That's where the creative development is. And so that's exactly what we want to get at. We want to get at the second category of things in development. Another way of thinking about it is genetics provides the architecture and the learning rule. Experience provides the learning and the plasticity. And I think by longitudinally looking physiologically at animals during development, that's the way to start parsing out the difference, how genetics and experience work together to create general motor intelligence. Okay, to make it a little bit more concrete, let's look again at this schematic. This is from a paper from Peter Strick's lab, but the ideas are due to Kenji Doya and many others. The idea is that the different parts of the brain, in particular the basal ganglia, cerebellum, and cortex, each rely on different types, different classes of learning rules. So for example, the proposal is that cerebral cortex relies on unsupervised learning, whereas the cerebellum uses supervised learning. But many of you probably know there was a recent paper, by Tim Lillicrap and Jeff Hinton, where they argued that the structure of cortical layers and the nature of cortical feedback connections might be designed specifically to implement backpropagation, which is like the bread and butter of supervised learning rules. So obviously, this is still an open question. So let me make this even more concrete with one more example. This is an example that is near to my heart. It's something that we studied a lot in my lab at UCSF. And this is the question of sensory integration in the posterior parietal cortex. So we know that vision and proprioception come together in the posterior parietal cortex to guide movement control. And we know that this is a highly flexible kind of integration. There's the Bayesian rules come into play. We know that this is highly adaptive. And for example, we proposed in my lab unsupervised learning models that could account for this kind of learning. So, maybe this is a good example of a cortical unsupervised learning system. But what we'd like to do is actually go and record this during development. And there's time to do this. In monkey, for example, it takes weeks for this kind of sensory integration to come online. And so I think that there is an opportunity to record these signals online during development and try to parse out what the learning rules are. And I've got one more example, but I will stop there because I'm out of time.
Okay, thanks very much, Flip. Let's see, we have some questions lined up. First one will be from Amy Bastian.
Hi, Flip.
Hey,
I liked your talk.
Thanks.
It was very good. I'm curious about, this is, I think, an obvious question coming from me, but I always wonder about like large scale recording and then just having free behavior. Are you proposing free behavior and somehow making sense of the relationship between these recordings, or are you proposing to actually have some behavioral constraints or look for specific things?
Yeah,
integration task is an obvious one. So yeah.
Yeah, it's a great question. And I've always been a little bit skeptical of looking at free behavior. And I've always thought that the right approach is to combine free behavior with sort of the kinds of psychophysical experiments that you and I and many others have designed for years that really get at specific questions. That said, I think that if you have enough data, and Chethan really laid this out nicely, I think in some ways, single trial data has gotten pretty far. And I think there is a lot that you can do with free behavior, but there may be a middle ground. You're not going to train neonatal animals to sit and do 1,000 tasks a day. But what you can do, a little bit like what the International Brain Lab is doing by setting up a specific experiment that everybody does, is you can set up specific kinds of devices, games, toys, environments that animals can are reared in, and that promote specific kinds of behaviors and interactions with those games, with those toys. And I think actually clever design of those sorts of things could go a long way to getting close to the kinds of psychophysical experiments we like in an environment that's really very self-directed. So I think the right answer is a combination, because that gives you both the broad, rich data set of natural behavior and the constrained data.
Thanks. I agree.
Okay, next question is from Abby Person.
Hi, Flip. You alluded to goals in your talk, and I was wondering if you could talk a little bit more about how you think goals may develop over the course of age, and how those goals interface with these motor control structures.
Yeah, that is such a deeply interesting question, and the short answer is I have no idea. But that said, it's clear that they do, right? A developing animal, one of its big goals is to just be near its mother, maybe to feed. As we get older, our goals get more sophisticated and more complex. There are probably some goals that are really foundational, like, for example, energetic type goals. And so I think this is exactly the kind of question that you can address by looking in more detail in animal models during development. And the experiment that I didn't have time to talk about was one of my favorite old experiments by Held and Hein, where they manipulate rearing in cats and talk about the difference between they show the need for sensory motor integration to get the kinds of posterior parietal cortex development. But the point there is that with animal models, there's the ability to within ethical constraints, of course, there's the ability to really control the things that are available and the things that an animal can choose to do. And in that way, I think we can even start getting at these very higher level cognitive questions like goals that really do play directly down into the low level of learning rules. So great question, exactly the kind of thing I think we'd like to look at.
Okay, next question from Andrew.
Hey, Flip, awesome talk. I had a question that sort of follows up on Amy's, and that is, you seem to be suggesting that just observing those neurons will take you most of the way, if not all of the way there. And I'm just curious whether or not you have thought about interventions, for example, in the plant or in the nervous system. I mean, obviously you have, but it seems like a real opportunity in the context of development where you have these particular milestones. Then you can manipulate and see how the system rebounds from that. Could you maybe just talk to how that would let you infer or take a look at the underlying learning rules?
Yeah, that's a great question. And I thought about talking about that, but there was already too much in here. And in any case, I think, this is where we are in neuroscience right now. We've got the tools to do these kinds of interventions, and it's really powerful. But I think what I would want to say is, look, you have to start at the beginning. And here the intervention is development. And so I think you have to back up, do the recording experiments. There's already enough here to be done to combine the kinds of sophisticated behavioral studies that Amy was talking about with these very complicated physiological experiments, get a baseline for what we're looking at. And then, of course, at some point, you are going to want to do these perturbations. And in some ways, like the old Held and Hein experiment, all these, like, sewing eyelids shut to study the course of visual development, those are perturbations. They're just perturbations on the input side. So I think, yes, you could do it in more subtle and interesting ways. You could look at how the response to simple plasticity driving perturbations changes as a function of age. All of that's true, and that might be the best way actually to really get at learning rules. But I think the first step is to just do the recording experiments.
I like the idea that the development is the intervention. I think that's cool. Thanks.
Okay, thanks very much for everybody who asked questions, and thanks very much for Flip. We will move on to our next talk, which is from Lena Ting. So Lena is a Portman Professor of Biomedical Engineering at both Emory and Georgia Tech. She's also the co-director of not one, but two centers for bioengineering at Emory, Georgia Tech. Her research focuses really broadly on how neural control is both instantiated and also constrained and shaped by the mechanics of the body. And take it away, Lena.
Okay, thank you. I'm trying to see all of you people. Okay, gotcha. Well, thank you very much for this wonderful symposium and giving me a chance to speak. It's been a great setup with the previous speakers for what I'm about to say. I'm hopping between my slides. So my talk is entitled The Brain and Body Compute Together, which I developed for the Brain and Computation Bootcamp when I was in residence at the Science Institute for the Theory of Computing. And I've since expanded that idea to expand across all of the sort of systems and hierarchies in the nervous system. And so I really mashed together what I put in the boot camp and what I took away from that really wonderful workshop and period of time. So many of you are probably familiar with Dan Walpert's explanation of why our brains are here to control movement, and this very simple example in a tunicate, a sea squirt, where when it has to move and sense the environment, it has.
Hey, I'm very sorry to interrupt you, but I think your screen is not actually being sent out. I think Flips is still sharing.
Oh, it says I'm screen sharing.
I'm getting these messages coming in. Others on the
it's sorry, that was me.
Sorry,
We're
okay,
good.
no, I apologize. Please go ahead.
Are we good?
Yes, we are good. Sorry.
Do people see my screen? Okay, so I think this example is really cute, but I study humans, and really to think about what does it mean that every brain function really is for us to move means that mood, motor, and mental functions are really not dissociable when we study movement. So you need emotions and autonomic responses to interpret sensory cues, cognitive functions to weigh cost and criteria, and to adapt to behavioral contexts. And this will trickle down into every aspect of the nervous system, even changing the way that we coordinate our body parts and our movements while we interact with the environment. And Dan has demonstrated this really nice in this work that's demonstrating these sensory motor prediction disorders in people with schizophrenia. And I like to refer to this as the schizophrenics can tickle themselves talk. But, you know, I started in neurosciences, not a neuroscientist, but a biomechanist, and I study balance control primarily in humans, but with lots of arms in computation and different animal models and simulation. So how do I even think about that? It was sort of a nice thing that I stored away, and I was like, okay, I can't deal with that. I'm just dealing with Newtonian mechanics, musculoskeletal systems. We started looking at feedback control in the spinal cord and the brainstem. And then as we studied the system, we have to study multisensory integration. And now we're studying perceptual responses and cortical responses by measuring EEG. So we've walked around this balance control problem. And as we started to study motor disorders, you cannot ignore the fact that the mood, motor, and mental functions are not dissociable. So we started studying people with Parkinson's disease, and then we wanted to look at brain activity during balance, which is normally an automatic behavior, but that can become controlled in a sort of top-down cortical way as people age, and in particular as they get Parkinson's disease. And so there are many, many examples of these comorbidities between movement disorders, mood disorders, and cognitive disorders. And this is just a set of examples here that I'm not going to belabor, but as I've tried to understand this system, I have a new vocabulary where I have to talk about anxiety. Are people anxious when they come in the lab? How is their cognitive function? Do they feel threatened? Do they think that what we're doing to them is an error, like if they're freaking out on this platform where we're moving them? Or do they think it's a fun carnival ride? And so, you know, wow, that's not what I was trained in at all. And so how do we understand these pervasive interactions between mood, motor, and mental functions in motor control? And I'm going meta, but I promise that I'll get down to nuts and bolts by the end of this and some things that we have tried. Okay, so why first of all, why is it that the brain and body and environment have to compute together to control movement? Now, while I was at the Simons Institute, there was a simultaneous session going on real-time decision-making. And Richard Murray, who's a control theorist at Caltech, gave a really nice set of talks, and I encourage you to go look them up because they're archived. And so he talked about control in the traditional view and in the emerging view. And these guys were talking about things like energy systems and bike shares and earthquake detection. But you know, what really struck me is that, okay, controls, like, it's not just feedback control for stability and performance and robustness, but moving towards this idea of this interconnection and feedback and communications all over the place, sort of embracing the level of complexity that we see in the brain. But he fundamentally referred to control as design of dynamics. And dynamics is movement. I mean, this is a slide I stole from him, but he basically pointed out some principles that feedback is a tool for managing uncertainty, feedback and feed-forward design dynamics, and that these feedbacks provide modularity and what they call interoperability to manage complexity. And interoperability is this engineering word for what we would call generalizability across a nervous system. Like, we want to use the same components to do different things from different systems. So he showed this cool diagram where he said, okay, in engineers, you have regulation, right? And I thought, oh, that's like the spinal cord and the optimization. Oh, well, we see that all over the cortices and brainstem. And then decision-making, maybe, you know, there's these prefrontal decision-making cortices, but one really interesting thing is that there's different math that explain all of these things. And I said, okay, can we talk, Richard? I think that maybe there's some parallels in what you're presenting. And he said, well, you know, I said, how do these things interact? And he said, well, actually, we engineer them so that they operate at a factor of 10 times different. And I said, well, what if we had a loop time of a factor of 2, just like we see with a short latency, long latency, and cortical response? And he just went, nope, nope, nope. You cannot have independence of these systems. They have to be very, very integrated. And so that just meant we cannot study any of these systems in isolation of each other. Okay, so what does that mean for us? It means that we really have to pay attention to all of these movements and the flexibility of neuromechanical control. So there's not just a way we move. There are many different ways, and we can see that there are shifts in the locus of control with disease, learning, emotion, and attention, which means there are very, very precise interactions between cortical control of movement and automatic control of movement, which is what we're studying in the context of balance control, and how those things might change with context, threat, which then release neuromodulators, the chemical kind that alter the function of various reflexes and central pattern generators and motor neuron inputs and outputs. So you need to study this cooperation across many levels, as Flip was talking about. And what that means is, I think that these top-down and bottom-up information flow is really not separable. We really can't talk about one thing for going one way to the other, but really being interactive. And I think the reason to do that is to be able to predict and to control dynamics. And so there's a great paper by Art Quo showing that central pattern generators that we see in spinal cord could maybe be thought of as predictors of motor dynamics. And even we're studying muscle spindles, this peripheral sensor, which really we cannot dissociate the sort of motor commands going to the system from alpha and beta motor neurons, and gamma motor neurons, and the environmental interactions and contractile mechanics, and even generating these muscle spindle sensory signals. So what do we do? How my big question is, how can these dynamics be predicted in a biologically plausible neural systems? And I met Sophie Deneve at the Simons Institute, and I can't go through all of this, but they're efficient spiking networks that basically allow us to design a network that implicitly solves an optimization problem. And we talk about optimization problems all the time in motor control. And it also moves a signal from a higher level to a lower level over the time course of learning, as we might assume works during skill learning and development. And the cool thing is that this system, this is a very simple example of two neurons trying to generate a particular output, and they interact with each other because of these sort of inhibitory interactions between the neurons, where it behaves like an optimal feedback controller, which is a way that the kind of math that we've used to describe motor systems at many, many levels, as sort of highlighted in this Todorov review. I don't know how much time I have, but Brianna Karpowitz, who did a rotation in my lab, based off some work I started with Sophie, just to try and say, well, what does this look like in a motor context? And can we make a simple motor unit recruitment model where all of the neurons inhibit themselves in sort of a Renshaw-like fashion, and that we sort of bake in an optimization, an objective function, and see what happens. And what happens is you get sort of some nice recruitment of motor units by the size principle, signal dependent noise. We kind of put that in the system, but emerged out asymmetric recruitment and de-recruitment of the motor neurons as you generate this ramping up and down force. Now, the really cool thing that makes me want to look at this is when you kill or silence a bunch of neurons, it generates the force, but the patterns of firing across these neurons is extremely difficult. And as someone who studies neural injury and degeneration, I think this is a really important adaptive robustness feature of motor systems that we really don't have any tools to explain right now. And the way that the network's set up, we could imagine how Renshaw cells could be activated by descending commands about how the neuron properties might be changed by neurotransmitters with various attentional and emotional states. And so, I guess, even if we could collect all of the movement data and the observed neural activity, we still need theories to explain how these sort of ubiquitous motor properties that we can describe by math, like really emerge from neuromechanical systems in a biologically plausible spiking with mechanics way. And I'm just going to end with sort of like, can this spiking network, a bunch of modules, coupled with mechanics, explain a lot of these features of movements that we've been talking about through all of the talks, from modularity, optimality, variability, development, and sort of mechanisms throughout the nervous system to predict dynamics. And with that, I'm gonna stop.
Okay, thanks very much, Lena. That was great. So I actually have a question. So you, in the first part of your talk, you were discussing how the brain and body compute together. That really reminded me of Abby Person's talk in the first half when she was talking about intermittent control and underactuation. And I guess what I'd like is, what do you think is the qualitative relationship between, the brain and the body computing together and intermittent control? So a super simple version is just when the muscles are off, all the computation happens in the body and the brain can take a break.
The muscles aren't always off.
But that can't be right for various.
No, but it's a really good question. And one of the examples I give is when people feel threatened or uncertain, like control moves simultaneously to the cortex and to the muscles. And that's what she's explaining. Like, we're starting to pay attention to things. And then what we do is co-contract our muscles, and then our muscles do the computation to stabilize ourselves. And it costs a little bit more energetically. And so that muscle stiffness allows you to use the longer loop time to the cortex to be able to make the moment by moment decisions. So you can't do cortical control if you don't also have very good stability at the level of the musculoskeletal system.
Terrific. So maybe another way to put it is that, you know, if, let's say you're really doing intermittent control and your control signals are super sparse, what you're saying is that you're actually setting up, you know, dynamic, even if you're not, you know, even if the EMG is silent, you've set up, you know, via the physics of the body.
Right.
Right.
And on the other hand, if you're highly skilled, we know that control moves subcortically, and people can be more efficient then because they've got a more precise repertoire of movements, and they're able to sort of respond more quickly. Once you start, I mean, the Amy showed this, once you start thinking about the movement, it just, you know, all of that goes to crap.
Okay, let's see, we have a few questions from the Zoom. Next is from, sorry, I've lost track. Someone can just jump in, I lost my note. Actually, Abby, are you up?
Yeah, Lena, I loved all that. It was a more real version of what I was kind of thinking about. So in one of your slides, you mentioned that the command is actually difficult to decode. And I found that an interesting comment, because I think one of the puzzles that I think those of us who study the central nervous system face with this idea of intermittent control is, what would that even look like? So with Chethan's beautiful state space dynamics, you know, there's this idea of this continuous representation of a reach or something like that. But I think that ultimately, you know, the command is sparse and it sort of kicks things off. How would we find it? Do you have insights into like what the structure even would look like?
So I think you could, so this is where I think some like computational, sort of theoretical computational models can help us kind of understand what are the potential scenarios and what would they look like. So in that last paper I didn't have time to talk about by Alemi, what they did is they showed that if you have 100 neurons that's learning some dynamics, that you get like a lot of burstiness, like the correlations across neurons. But if you increase the number of neurons, then they start to look decorrelated and asynchronous when they're receiving the same motor command. So we really have to think about what are the structures and sort of relative amounts of inhibition and excitation, and how far spread they are. And Sophie, like, she has specific predictions about how many Renshaw cells, if they project to, you know, above 20 motor neurons, like whether you'd predict synchronous or asynchronous behavior. And it turns out the number is something around 15. So that's really sitting on the edge of synchronous and asynchronous.
Okay, great. Here's a question from Slack. This is from Sean O'Connell. Could fluctuations in concentrations of neuromodulators induce changes in state space dynamics within cortex, spine, et cetera? And, you know, I'll add on top of that, you know, maybe relating to some of the, you know, mental influences on motor control you were talking about earlier.
So I didn't quite understand the question. Could these?
Could changes in neuromodulators induce changes in the state space dynamics, you know, in any of the multi-levels, you know, cortex, spinal cord, et cetera? Could that, you know, my elaboration on it, could that be responsible for some of the, you know, you talked about these sort of extra motor influences on motor output?
Sure. I mean, I think that there are many ways in which we could do that. We made a very, very simple toy model. So, and the types of things that can begin to simulate are what are the effects of sort of diffuse neuromodulation, as C.J. Heckman puts it, and then focused local inhibition that's related to the task. And so it should be able to jack up the whole system.
Okay. All right, so thanks very much, Lena. Our next speaker is Amy Orsborn from the University of Washington, where, sorry, let me get to my notes. Where Amy is a Claire Booth Luce Assistant Professor in the Department of Electrical and Computer Engineering and Bioengineering, and she will be telling us about network level distributed motor learning.
Great, thank you. Hold on one sec. All the Zoom controls are making it hard for me to start the slideshow. Great. Okay, so thank you very much. Really excited to be here. This has been a fantastic discussion. So one of the hallmarks of the motor system is that it's flexible. And we've recently all been reminded of this now that we're pressing elevator buttons with our elbows rather than our fingers, thanks to COVID. And the motor system is fantastic at this, sort of quickly and rapidly adapting things that we already know how to do to new circumstances or adding entirely new skills. And I'm really interested in understanding how the motor system achieves this flexibility and the role of cortical areas in that flexible computation. And the reason I focus on this is because if we understand how the natural motor system is so flexible, we can then actually engineer the system through neural interfaces to restore function in the case of injury. So when we look at motor learning, what is very clear at the level of behavior is that it's not actually one thing. There's multiple co-occurring learning mechanisms that all together contribute and give rise to the flexible behaviors that we see. When we think about how that's implemented in the brain, we of course know a lot about basic mechanisms of synaptic plasticity, but largely understood at the scale of pairs of neurons. And how the large distributed networks that we've heard about today that are controlling our motor behaviors actually implement this learning is a lot less clear and is now becoming a real focus. And I think we really need to tackle this network scale of problems if we're ever going to be able to bridge from biological implementation towards these distributed computations that we see in the level of behavior. So networks, of course, are really difficult to grapple with. And let's just take a toy example of two cortical areas, premotor cortex and primary motor cortex, where primary motor cortex through corticomotor neuronal cells are sending projections down to muscles to control behavior. If we see changes in the behavior, it of course could be due to local plasticity in M1 specifically, or maybe it's actually plasticity that's happening upstream in premotor areas, or even further upstream in an area that we're not recording from right now. Or maybe it's a specific plasticity that's targeting the projections and how the two areas communicate. In addition to this sort of distributed anatomy, we also have potentially distributed computation playing a role. So there have been some recent really interesting papers showing that if you record from large populations of activity across different cortical areas, that there are potentially population level mechanisms that give rise to really flexible communication strategies. And those might be really important for these really rapid hairpin turns that you see in motor learning. So when we think about trying to untangle all of these different things that are happening in these networks, we come up with a really big wish list. We need to know the output layer to some extent. We heard earlier about how it's really important to understand the inputs of the system. We also need to understand the outputs if we're ever going to understand the computation happening in between. We also need to be able to measure the networks themselves, which is really challenging. And we need to be able to do that both really quickly and longitudinally, because motor learning spans timescales of milliseconds to months. So on the first challenge of knowing the inputs and the outputs, I would argue that we to some extent have a lot of the tools that we need, and that's provided through neural interface technology. So sensory motor neural interfaces, where you directly read out activity and use it to control an external device, define novel sensory motor transforms, and allow us to actually sort of fully define the sensory motor loop. And so all of these reviews that I'll point you to for details kind of highlight some of the advances in these approaches. And this figure kind of diagrams all of the different manipulations that have been done to really probe these systems. And what all of this research really highlights to me is actually evidence that neural interfaces engage multiple forms of co-occurring learning mechanisms, much like what we see in natural learning. So this gives us a test bed for thinking about kind of studying distributed learning mechanisms. So now let's think about how we record the networks themselves. And so we, of course, as we heard from Flip, need to basically record from everywhere, right? And we have emerging technologies like neuropixels or the neural matrix array, where we can record from thousands of electrodes simultaneously. We also have imaging techniques where we can start to record from the whole brain of at least small animals. And so there's a clear path forward to some extent, but as Flip kind of alluded to, there's always scaling trade-offs with these techniques when you think about really big brains and big networks. You're always trading off between spatial and temporal resolution or the density of your measurements versus coverage of the brain as a whole. So in addition to just measuring the activity, we also ultimately need to get at the underlying network structure as well. And we need to be able to measure that network in a dynamic way. So let's think about our, and here's where I think our existing techniques sort of leave me at a little bit of a loss of exactly what the path forward is. So if we think about our techniques for measuring large-scale functional connectivity, where we want to do causal manipulations and not just try to infer connectivity from statistics of the populations, the usual approach is that you stimulate in one site in your network and then you measure all of the responses elsewhere, and then you gradually work through all of the nodes in your network. If you think about how this scales though, of course, this doesn't work very well when you start getting to big networks because it takes a really long time. And so you start losing all of the dynamics of the network. Similarly, we have challenges when you think about measuring the anatomy as well, where we have techniques where you can do really beautiful high resolution dynamics at a really small scale of sort of synaptic processes. Or we can do whole brain beautiful tracing with things like clarity, but it's the definition of static because it's ex vivo, right? And so there's a big gap, I think, and when I think about my wish list, what I really want is methods for rapid network identification in vivo for really large networks. So both of these, the recording and the network ID, really point out this problem of scaling, right? And what we're grappling with, kind of to put it really simple, is that brains are really, really big. And that's particularly true if you care about human brains or primate brains like I do. And so one potential strategy for dealing with this, rather than trying to actually record from everywhere, is to do targeted data-driven network-based sampling. So you can think of the analogy that we need kind of a whole map that we can then selectively zoom into. And so this motivates a goal for kind of multi-scale data integration, where you have different scales of data that you can combine together and mesh both over time and across spatial scales to basically do this zooming and panning. And I am by no means the first person to point this out, and that's in fact kind of a lot of what people are starting to achieve in the imaging world with these mesoscopic imaging approaches. So I don't want to at all suggest that we've solved this problem, but I did want to kind of point out some approaches that I developed while I was a postdoc with Bijan Pesseron with sort of this bigger picture vision of studying large-scale cortical networks in non-human primates. So we focused on developing an implant system that was sort of really modular and flexible, where we could kind of do whatever, a wide variety of different combinations of techniques. And so this lets us do electrophysiology and also imaging of at least multiple cortical areas simultaneously. We can combine this with optogenetics plus electrophysiology to start looking at connectivity, and we can do multi-scale electrophysiology towards this goal of sort of integrating across scales and targeting networks. So we, of course, the final piece I want to comment on is that ideally we'd be able to do all of this measuring large-scale networks, not just quickly, but also for long periods of time. And that's because, and just to think about studying learning, task-related learning occurs both while you're awake and while you're asleep. And skills ultimately are learned on the order of weeks to months. And so when you start thinking about that, what you really need to do is basically record 24/7 for really long periods of time. And in the Passeron lab, I started doing these types of experiments. We have the technology through wireless recordings, for instance, to start being able to approach 24/7 recording. But the thing that this kind of opens up, and where I think Chethan and I should probably talk more, is that back of the envelope calculations show that you get to petabyte scales of data really, really quickly. And so what you need this opens up need for kind of computational and statistical frameworks, data infrastructure, and pipelines. And so I'll wrap up here by just saying that I spend a lot of time looking at this diagram from a recent Krakauer review of motor learning. It shows that even the simplest behaviors that we try to make simple in the lab actually involve really distributed learning phenomena. And if we're ever going to link those back to biology, we really need to start thinking about the tools to measure and interpret learning in networks. So with that, I'll thank you and point you to where you can find us on the web.
Okay, thanks very much for a great talk. And let's see, Abby Person, go ahead.
Amy, that's very exciting. I am super interested in what you're looking for during sleep, because I think that is a completely underexplored part of motor consolidation and learning. There are stories and tales in the song system where there's, sort of kick down, drag out fights about what people are looking at in sleep. And I'm wondering if you have insights about how you would interpret what kinds of patterns are observed in sleep and what they mean.
Right, that's a really fantastic question. And I agree, I would say, a pretty open space. I don't have excessively strong hypotheses. The thing that I think, so the paper that I cited there is a pointer to some really great work out of Karnesh Ganguly's lab, where they leveraged actually brain-machine interfaces where we know exactly which output neurons are driving behavior. And they've done some really nice studies where they then try to look for evidence of this kind of consolidation process, right, that's happening between the active practice and then during sleep. And so I think those are the types of questions that I would be most interested in, is understanding kind of how the rapid short time scale learning phenomena that we see, right, during practice get consolidated, and in particular kind of around these credit assignment problems, right, of which neurons are driving behavior.
Okay, next we have a question from Andrew.
Yeah, so I'm just noting this contrast. So in my talk, for example, I'm like, oh, you really have to figure out what this one little neuron in the skin may or may not be doing, or else you're like toast. And then in your talk, and Philip's talk and Chethan's talk, you're like, don't worry about it. I'm just going to record from every neuron and it'll be all good. And I'm just wondering if you could speak to that tension between those approaches.
Yeah, it's a great question, and I think I completely agree. I think that I would try to position myself in the middle, in the sense that I agree that it's, the full let's record absolutely everything, we'll figure it out on the back end, I think is maybe unlikely to be. I have my doubts. I think the idea for me in particular, and the motivation of using these neural interfaces as a strategy, is that it strips down the system in a slightly different way, right? That then starts to create circuits that are a little bit more tractable, that then hopefully we can, this approach of kind of record everything potentially would be useful.
Okay, here's a question from Jerry Fishbach. Have you ever looked at sharp wave ripples in the motor system as being a potential mechanism of recall?
Yeah, that's a great question. I have not yet. I've been sort of, it's taking us quite a long time to actually dig through some of the sleep data that I've been collecting. But I know that, again, pointing back to Karnesh Ganguly, whose work I'm pretty familiar with, he has certainly seen evidence that some of these oscillations are really critical for that consolidation process.
Okay, thanks. Next question from Flip.
Hey, I'm going to start with a comment, Andrew. Your question was a good one, and I think there could be a whole symposium just on that. Now, Amy, great talk, really, really good stuff. I'm deeply intrigued about the 24/7 data, and I'm going to turn Amy Bastian's question to me back on you, which is, now that you actually have those data, has there been an ability yet to, or have you looked yet at relating what you see in careful, well-designed tasks to natural behavior? Yeah,
that's a really fantastic question, and the short answer is that I don't think we yet have the, to be totally honest, I don't think we have exactly the right frameworks for doing that quite yet. And in part because, you know, the 24/7 data is really difficult to parse, right? You don't, you know, this is where, again, like Chethan's tools are fantastic for single trial analysis because you don't have trials, right? It's just completely unstructured data, and trying to understand what are the relevant events, I think, are really difficult. So one thing that we've started doing, that I've started doing, I'm collaborating here with Professor Ellie Schlizzerman, who does a lot of work on trying to develop unsupervised methods for in machine learning for mining data. And I think that potentially those types of computational approaches will actually start to reveal some of the structure, right, that we're potentially seeing, that'll then give us an entry point to trying to link things back together across these totally different contexts.
Thanks.
Okay, thanks very much. We will then move on to Rui Costa, who's the final speaker in our group. Let me bring this back up. So Rui is professor of neuroscience and neurology at Columbia University, and he's also the director and CEO of Columbia's Zuckerman Institute. His work combines behavioral, genetic, and imaging methods to look at motor control and motor exploration. So take it away, Rui.
Hello, can everyone hear me and see the slides? Good. So first, I would like to start by echoing what Chethan and other colleagues said here. These are tough times where it's difficult to imagine what's the role of science. But I wanted to say that I think we have a role in fighting systemic racism, bias, lack of inclusion, and the people that are present just in this symposium and this consortium highlight the importance of diversity of thought and what it brings, not only as the right thing to do, but the greatest way that humanity can achieve their goals. Anyway, now on to the science of what the questions that I'm gonna put today. So for most of body movements, muscles are activated by spinal cord circuits that have some higher hardwired logic. Those circuits receive descending input from specific populations in the brainstem, and that has been quite well studied by a few people, or is being studied now by people like Sylvia Arbour. We have focused more on what other brain areas, for example, the basal ganglia, how they modulate that descending input. But today I would like to focus more about this type of descending input here from motor cortex, the direct corticospinal projections, and to which we know perhaps, for which we know perhaps less, and also the fact that these projections broadcast to many regions. So, for example, if different brainstem populations, here depicted in different colors, target specific spinal circuits, do different populations of motor cortex neurons target different spinal circuits? Also, what is the relationship between the activity of these neurons and the activity of muscles? And how does the spinal cord transform these brain signals or help the transformation into muscle? What's the biological but also computational understanding of this? How would it look like? And if we understand it, can we mimic it in a meaningful way or perturb it? And so here, just to exemplify, in the mouse, and this is work from Samaher Fajri and Anders Nelson, if you inject a retrograde virus in the cervical cord from which the forelimb motor neurons emerge, this is the extent of where the cell bodies are, the extent of the corticospinal network. And I hope many of you appreciate that it's quite large, and maybe and it spans many areas, not only the motor cortex. But even when you go to motor cortex, you can see that if you jump back from the spinal cord with a Krieger virus and then complement with a reporter virus in motor cortex, you see that those corticospinal neurons project, send collaterals throughout the brain, a lot to basal ganglia, to striatum, but through many structures until they reach the spinal cord. So maybe we need to understand what is happening here to understand cortical control of movement. And so one interesting thing just to highlight, for example, if you use retrograde virus to jump back from CHOX-10 interneurons, which are premotor interneurons, versus somatostatin positive interneurons, which are a mechanosensory interneuron, you see that the locations of the cell bodies in the cortex are in quite different positions, highlighting that there are indeed different corticospinal populations. I mean, this is just an example, but if we'd have access to much more detail, maybe we'd know more, or maybe not, because look at this interesting thing. If we look at the activity of these two types of populations of neurons, and this is with imaging, when a mouse is moving just a wheel, an actuator, they seem to be doing the same thing. So motor cortex, or these corticospinal neurons, despite the fact that they are targeting different spinal interneurons, may be conveying similar information, or we need better tasks to get at it. So going on what, for example, Flip said, maybe we need a type of IBL, but where we have actually multiple tasks. And here are several tasks, for example, that we've been trying to develop with this consortium in mice. This is a lever press task. This is a continuous 2D finding a target task. This is an isometric contraction task. So maybe these reveal surprising things. And then another important thing in terms of tools is the fact that for much of the time, our lab, for example, guilty as charged, has looked at the relationship between neural activity and, for example, the pressing the lever or moving of the actuator. And here you see, for example, some corticospinal cells that follow exactly the movement of a lever, but you see others that not necessarily are more on cells or off cells for a whole sequence of presses. But then when you look at the muscle activity, not just the actuator, you see that the complexity is quite interesting. In this case, what biceps and triceps are doing, alternating. And again, you see that there are cells like these ones, many cells with very strong correlation with muscle activity, but cells that have much less correlation with muscle activity. And this is with average muscle activity and bulk muscle activity. But with the tools in this consortium, these are data from Alice Mosberg and Bryce Chung, using the flexible high resolution EMG recordings that Sam mentioned. And these are single units recording in the trapezius muscle when an animal is, for example, in a reining wheel. So we have hope that tools like these could help us establish better what behavior is, what movement, but also relate the neural activity with muscle activity with computational tools like those of Chethan. But still, to get to the causal relationship between single cells in the cortex and muscle, we need better things. So maybe one way, and this is work by Vivek Kathali and Bastian Peterka, is to use stimulation of individual identified cells, here photostimulation, for example, of this single neuron versus a control cell, and try to use that to determine the relationship between the activity, the direct relationship of cells in the cortex and single units in the muscle. And finally, as Andy mentioned, we need tools to try to target the specific circuits in the spinal cord and see what is that transformation. And here's an example that David Ng developed, this new intersectional approach that instead of using the stop Cree, which is quite leaky, the more classical, he used a split Cree with the stop codon in the middle. And so, for example, here using Fox P1 and Batch Flippo with the intersectional Cree, he can target very specifically the lateral motor colon motor neurons. He's developing the same for Renshaw cells. So it is a very exciting time, and I think getting to what Flip mentioned, I think this international consortium with these amazing fellows here is one of these attempts to go beyond what we could have dreamt when we started discussing, where everyone threw ideas like these. I just put here the highlights of, I think, what we could get at if even some of these tools would work well. And I'd be glad to take questions. I know we're ready to go for lunch.
Well, I'll start with one, and this is maybe something I'll also sort of branch out to for all the speakers on Zoom and in the audience. So, okay, so really these are amazing tools. I mean, so you can identify different genetically, manipulate different genetically defined descending projections, and then with the kind of targeted photostimulation, we can do, you can manipulate the pattern of activity in a particular one of those. So I guess one thing, that I know we're all thinking about this is how do you map that onto tasks? So, it could well be that, the computational role of some descending population is going to be very different from one task to another. And, you're laying out a strategy, which I very much like, where you've got, mice doing 3 or 4, different, constrained but still really rich tasks. I mean, maybe this is a provocative question, but wouldn't it be better to do that with unconstrained behavior? I mean, what do you get from, you're sort of making a bet that one of your cell classes is going to map.
Exactly. So as you know, Sam, we put quite some effort on characterizing unconstrained behavior as well. The difficulty is, for example, how can we control for an issue of, for example, if a behavior is feedback controlled or feed forward controlled? Very tough. So then with, for example, this 2D task, we can move the starting position immediately, and we ask what strategy is the animal using. The same with isometric or co-contraction, right? So it's very hard to just wait that happens. So I would like, for discussion with Phil, I think without multiple tasks. So, we always think single task, multiple approaches. But our tool, what we want to understand is behavior. It's the behavior of the body and muscles together. So part of our questioning tool are the behavioral tasks, right? Because we may get to a point where, Sam, you say, we can't disambiguate between 2 hypotheses, and we may need a circuit tool, a computational tool, a recording tool, but we may need a task as a tool, a new tool to interrogate. So that's what I was trying to get at, that without having the proper tasks and just measuring correlation, we may be fooled. I mean, we still may be fooled. Let me get it. Motor control is the most exciting thing, and the most complex thing out there. But that's just an attempt that, and I think you all agree, right? We need the multiple approaches, including the naturalistic.
Okay, thanks very much. So we're still open for other questions. I guess just one thing I'll mention, Rui, that you pointed out is that it hadn't occurred to me, this distinction between constrained and unconstrained and isolating feed forward and feedback. So earlier I might mention generalization is something that's great to study. There are very defined ways of studying that with constrained tasks, but you're also right that it's good for explicitly looking at the feed forward component of some task.
The task or feedback, yeah.
Okay, any other comments or questions? If not, I will thank everyone for coming. I want to especially thank the speakers for taking on was a brave and challenging assignment. And thank you very much for everybody who has been watching the broadcast. I'll also mention we will make a recording of the broadcast available shortly. It will be on the same YouTube channel. Oh, sorry, you will be on the same YouTube channel that you are watching this on now. And there is a question. This is from James Finley. To what extent do we or should we think about the spinal cord as a source of input-output from the brain versus a central component in the CNS networks that subserve learning and action?
So that's an exciting question, and I wanted to address it. I went too fast, but what I wanted to highlight when I mentioned that the two populations of corticospinal neurons seem to have, going down from motor cortex to different spinal interneurons, seem to be sending down similar information. It kind of pushes back the problem to the spinal cord. So in a way, I'm really glad about this question. It made us think, it would be, okay, we need to study everything up in the cortex, and the spinal cord just does some type of transformation, right? But the fact that the similar information is going to many of the neurons pushes a lot of the computation back to the cord. And now we are all shying from these. You saw I put in the last slide a very shy attempt to get at the spinal cord because of the ability to measure and manipulate. But the data are telling us we need to go back and understand, no, these neurons are getting similar information from the brain. They're just treating it differently based on other inputs and where they are, right? The mechanosensory interneuron is getting sensory input. And what is the cortex telling that? Andy's saying is telling you the sensory consequences of the goal or of the movement, and similarly, what is it telling the motor neurons and the other circuits? So this is an excellent question, and I think I wanted to point out that the measurement of single units is the first most exciting thing, because in fact we're measuring alpha motor neurons, and but alpha motor neurons directly, right? Those single spikes, and that's very exciting. At least we know one part of the output.
Okay, so thanks very much again to all the speakers and people in the audience. The recording will be posted shortly, and also please stay tuned for other announcements of consortium events. We will have other meetings, possibly similar to this one, and in the slightly longer term, we will be having training courses on the technologies that we are developing. So thanks very much, and have a good day.
Just like to add, before we quit, I'd like to add a couple more thanks. So I'd like to once again thank all the people from Emory TMLS, particularly Tira Ward and Amanda Jacobs from the Simons-Emory consortium here for just doing all this fantastic work so we actually get stuff off the ground. Also really like to thank all of our students and postdocs that have been hanging out on Slack, feeding us questions from YouTube, and all of you for making all the great comments on YouTube on the side. I've been following it sort of along, and it's been a really fascinating and entertaining conversation. So thanks again to all the speakers, and hopefully we'll see you again in a month. Bye.
Thanks, guys. That was awesome.
Thanks, guys.
Thanks, everyone.
Thanks, everyone.
Later, thanks.
Thank you. This was fun.